Good
Good
morning,
morning,
everyone.
everyone.
On behalf of the Harvard Undergraduate OpenBio Laboratory, welcome to Revolutionizing Life, Frontiers of Biotechnology. We're absolutely thrilled to have you here, whether you're joining us from Boston, across the country, or anywhere around the world.
Before we dive in, we'd love to quickly introduce ourselves. I'm Julia Torrey, a sophomore at Harvard studying the history of science.
And my name's Adie Selassie. I'm a junior at Harvard studying neuroscience.
We had the honor of organizing this year's conference, and we're so grateful to have you all here with us today. We're kicking off what promises to be a very exciting day and an incredible gathering of students. Over the next few hours, you'll have the opportunity to engage with some of the brightest students, researchers, and pioneers in biotech and biomedicine. Together, we'll explore emerging discoveries, powerful innovations, and the big questions shaping the future of life sciences. This is our third time hosting this conference, and each year it becomes more dynamic, more inspiring, and more community driven. What makes this event truly special isn't just the remarkable speakers or the groundbreaking ideas, it's all of you. Students, researchers, and innovators coming together to imagine what's next for biology and medicine.
We're deeply honored to welcome two visionary keynote speakers. This morning, you'll hear from Dr. Omid Farokhzad, founder and former director of the Center for Nanomedicine at Brigham and Women's Hospital, and now the CEO of Seer, a company reimagining proteomics and biological discovery. And after lunch, you'll hear from Dr. Ed Boyden, the Eva Tan Professor in Neurotechnology at MIT, and a leading innovator in brain research and neuroengineering. We also have a phenomenal lineup of interactive workshops led by student groups from the Harvard Biotechnology Club, Stanford Biome, NYU iGEM, and Cornell iGEM. These sessions are designed not to just inform, but to spark curiosity, inspire new ideas, and encourage meaningful conversations.
In the afternoon, we'll hear from the Harvard Grid Accelerator, an organization that supports the launch of groundbreaking biotech and biomedical engineering ventures. They're going to share pitches from some of the exciting projects that are currently in development. And later today, we'll also host a virtual poster session, which features original student research. You'll have the opportunity to ask questions, dive into exciting projects, and connect with a global community of future scientists and change makers.
We are beyond excited for what's ahead, and most of all, we're thankful that you're here with us. And now, to kick things off and give you a closer look at the heart of this organization, we'd like to pass it off to the presidents of OpenBio, Sean and Hugh, who will share a few words about our mission, our origins, and the work we do year-round. So let's get started. And once again, welcome to Revolutionizing Life.
Good day, everyone, and welcome. It is so nice to see you all here, and we're excited for the fun day to come. To those of you joining us from the United States and the rest of the Americas, thank you for waking up early to learn about biology. And to our global attendees, we appreciate you taking the time out of your day to join us.
We are Sean Mang and Hugh Hankinson, the co-presidents of OpenBio, and we are so thrilled to be hosting you today and for the third iteration of OpenBio's annual conference. Before getting into it all, we wanted to open today's discussion by telling you a little bit more about who we are and what we seek to do.
Just 3 years ago, the OpenBio Laboratory was launched here at Harvard with the mission to empower student biology and to build communities at Harvard, in Boston, and around the world that are dedicated to using biology to move our world forward. Within our operation, we support student-led biology research by providing resources, funding, lab space, and advising to help undergraduates turn their research visions into a reality. We seek to make exciting and meaningful biology research accessible to all, and our team is committed to increasing access to research opportunities for underrepresented groups in science in all of our activities.
Yeah, we view the flourishing biology revolution as the next great saga in scientific research and technological innovation. Already, we see the benefits of this movement are becoming clear, with the development of new therapeutics, advances in agriculture, and innovation in energy and materials. Throughout today's event, the theme of which is revolutionizing life, we will hear from scientists, innovators, and students at the forefront of the field. Their work spans everything from tools to map the brain, multi-omic analyses to exoskeletons, disease diagnostics, and technologies enhancing movement and accessibility.
Despite the potential that biology holds to make the lives of billions around the globe better, in the United States at least, our ability to do so, to do biology research, to do biotechnology, is being severely limited right now. Over the past half century, the National Institutes of Health have played a pivotal role in shaping and supporting biomedical research across the country. Federal research dollars have been essential in developing novel understandings of the human body, of disease, and of healing, and have heavily influenced dozens and dozens of therapeutics brought to market worldwide. With promised reductions in NIH expenditures, and it is inevitable that research in the United States and across the rest of the world will be limited in their capacity to further the life-changing work that they do. Unfortunately, as a result, many will go sick who might have otherwise been healthy, and many will die who might have otherwise lived.
Moreover, the targeted cancellations of grants supporting work into persistent disparities in health and nutrition, as well as those projects investigating how to make medicine more fair, equitable, and effective, represents an unmitigated attack on free speech, academic inquiry, and the dream of making the world better for some rather than just all. As scientists, researchers, inventors, and learners, each of us has a duty to stand up for biological research and to fight as hard as we can to innovate and investigate, despite the barriers we face. We have no doubt that today's speakers, their stories, and their work will reiterate to you the importance of biology and biomedical research in shaping our current world and the world of the future. On a logistical note, here's what to expect for the day. This right now is the welcome address at the top of the agenda there. We'll wrap up in just a second and move to our first keynote. From there, we'll lead into our workshop session. You'll receive a sheet containing the Zoom links. Please feel free to go to the session that interests you most. As Adi and Julia said, we have an exciting selection offered by our friends at Stanford Biome, Cornell iGEM, NYU iGEM, Harvard Undergraduate Biotechnology Group, and our own lab team. At 11:30, we'll break for lunch. Please be back on the webinar at noon for our second keynote. Following this, our panel session will begin at 1:15 and we will close with posters. And that's all on logistics.
To close our remarks today, we'd like to extend our sincere thank you to everyone from the OpenBio team for their work in organizing today's event, but especially to Adi and Julia. Thank you so much. The co-directors of the conference and the masterminds of all that you see today. We'd also like to recognize the OpenBio lab team for hosting an exciting workshop later this afternoon, the OpenBio finance team for sourcing the support that makes this all possible, as well as our speakers, all of whom are wonderful and we're excited to hear from, and our partner organizations across the country. Thank you all to you as well for being here, to being dedicated to learning a little bit more, and to taking the time out of a Saturday to try and understand the world a little bit better. Again, it is our absolute pleasure to welcome you to the start of Revolutionizing Life. We have no doubt that you'll learn a great deal and leave inspired to go forth and continue changing the world. It's now our pleasure to hand back off to Julia, who will be formally introducing today's first speaker in just a few minutes. Thank you very much, everyone.
We're just going to take a few seconds to let our first keynote speaker of the day join. We're running a little ahead of schedule. So just please wait patiently while we do that, and we'll be introducing him shortly. Thank you so much. I'm thrilled to introduce our first keynote speaker of the day, Dr. Omid Farokhzad. Dr. Farokhzad is the chair and chief executive officer of Seer, which is a company that is imagining and pioneering new ways to decode the secrets of the proteome to improve human health. Previously, he was a professor at Harvard Medical School and founded and directed the Center for Nanomedicine at Brigham and Women's Hospital. He has authored over 190 papers and is an inventor of over 250 issued and patents. Omid previously founded Bind Therapeutics, Selecta Biosciences, Tarvita Therapeutics, and Pronomic. Dr. Farokhzad is a 2023 Mustafa Prize and the 2013 Rusnanov Prize for his work on nanomaterial surface modification, as well as a 2018 Fellow of the National Academy of Inventors. He is a recipient of the 2016 Ellis Island Medal of Honor and the 2014 Golden Door Award from the International Institute of New England for his scientific, societal, and economic contributions to America as an immigrant. And he was named the 2012 Ernst & Young New England Entrepreneur of the Year Award. We are so honored to have the incredible and renowned Dr. Farokhzad speak with us today. Please join us in welcoming Dr. Farokhzad. I think he's going to be on in just a second.
Okay. Julia, can you hear me?
Yeah, we can hear you. Thank you so much for joining us. We're so grateful to have you here.
Pleasure, Julia, and thank you for inviting me, and super nice to see you. Just want to make sure that I'm able to share. So let me try this. Bear 1 second with me. I'm going to share my screen, and you let me if it's successful or not.
Will do.
Let's see here. Actually, let's try it this way. 1 sec. I'm going to do this. Hang on 1 second, actually, Julia. Let me do a presenter view. Julia, what are you able to see right now?
Just like a full screen of your slide. That's all. So yeah, it's perfect.
You just see the slide, yes?
Yes, just the slide, nothing else.
Perfect. Good. Okay. Well, then I think we can get started. So,
yes, amazing.
Okay. So, Julia, thank you so much. And real pleasure to be here. So I'm going to speak for about 30 minutes, hopefully, and leave some room for Q&A. And by way of background, I'm going to give a talk that initially starts off with some work that we did as in our academic labs that was at the Brigham, and then subsequently talk a little bit about the way it transitioned into some of the work that most recently going on in a company that I've now transitioned to and I work with, Seer, based in the Bay Area. Okay, so back to my academic days, always start with a financial disclosure, which is I've been a founder of a number of companies, and so some of the viewpoints I give does not represent those views. They're going to be my academic views, and then the second half of the presentation that relates to Seer may include some forward-looking statements. And consistent with that, this is the safe harbor disclosure for Seer, which is a public company. Okay, so let me start off by a little bit of academic work, and of course, many thanks to the folks that are on this slide. These are the folks that did the work. I just have the pleasure and the honor of talking about it, but the heavy lifting is done by the people that you see here. By the way, I'm super proud of many of them. 18 of them are now professors. 2 at HMS. One of them, Wei Tao, actually holds the Farokhzad chair at the Brigham. So just, they've done amazing. All right. So we're going to talk about nanoparticles and different nanoparticles for various different biomedical applications. And nanoparticles can be engineered with vastly different biophysical chemical properties, and They can have different shapes. You can decorate their surface in a number of ways. They can be designed for various different applications, imaging, theranostics, therapeutics. And nanomaterials themselves can even have biological function that you could capitalize on. And the field really has exploded. I was fortunate uh, uh, to have was able to join the laboratory of Professor Robert Langer at MIT as a postdoc, and before that, I spent about 7 years during college and medical school training as a molecular biologist. But when I went to Professor Langer's lab, I didn't know anything about nanomaterials or chemical engineering, and this was in 2002. And back then, there were not a lot of papers published on nanoparticles. In fact, if you look at that chart in aggregate, through 2000, there were about 1,000 papers published on nanoparticles. But as of now, about 380,000 papers published, and it's just the field has been exploding. And I was really lucky to be kind of there right at the beginning of this revolution. Now, we're going to talk a lot also about the way nanoparticles interact with biological systems, um, and and this nano-bio interface is important, um, and folks really refer to the nanoparticle design as its physical-chemical, if you would, identity. But the moment a nanoparticle interacts with a biological system, a layer of biomacromolecules forms on its surface, and that is been referred to a protein corona, for example, in the case of protein coating on the surface of a nanoparticle. And this was an observation really that Kenneth Dawson made in Dublin, and in a paper that he published in 2007. Really, he coined the term protein corona. And the field of protein corona, as you can see, since Kenneth published this paper, has also been growing rapidly. Now, most of us were working in this space. We just never refer to it as protein corona. In fact, if you look at the academic literature, often people refer to this as biofouling, because we didn't want these macromolecules to bind. And when they did, we were trying to avoid that. And so we refer to it as fouling, but Kenneth actually said, look, at equilibrium, the physical chemical properties drives what binds, and then he coined the term protein corona. Okay, so what I'm not going to talk about is all the various different nanoparticles that we have made over the years. I think those are readily available in PubMed, and you can go take a look at them. But suffice it to say that we've worked on various different systems for targeting hybrid nucleic acid delivery, protein delivery, small molecule delivery, combination therapy, theranostics, and imaging, working with different nanomaterials, including inorganic systems like gold and iron oxide, and organic systems like polymers and lipids. But what I do want to talk about instead, if you don't mind, is a very specific use case and what we learn. And by the way, in science, it's not always linear forward. It's usually 2 steps forward, 1 step back, and you're always standing on the shoulder of the guy who came before you in terms of what they learn and what you build on it. So I'll highlight some of the contributions we made that built on the works that previous colleagues had done. Okay, so I was trained as a molecular biologist, and I was very fascinated by this class of ligands called aptamers, which are essentially single-stranded nucleic acid that can fold on themselves to create three-dimensional structure that can have binding properties analogous to antibodies, and they can have super high affinities, and they can be very specific. You can create aptamers that can distinguish, for example, between theophylline and caffeine, 2 small molecules that are very, very closely resemble each other. So before me, another scientist in Bob Langer's lab, Roxandra Greff, had developed dye block polymeric systems that were amphiphilic. It had a hydrophilic end and a hydrophobic end, and it could self-assemble in a nanoparticle where the surface was largely hydrophilic, and then the core was hydrophobic, and the core could encapsulate drugs, and the surface, by virtue of being hydrophilic, would allow the particles to actually circulate in the body for a long time. And that was work that was published in Science in 1994, long before I got there in 2002. But building on Roxandra's graph, the idea I had was to attach ligands on the surface of the nanoparticles to create targeted particles and then use the ligands that can target tumor antigens and specifically that are specifically expressed on cancer, but on normal, and to develop targeted system for drug delivery applications. And we built that. You can see that if you can put some red dye inside it, that it goes to cells that are intended to be targeted. These are prostate-specific membrane antigen positive cells, but it doesn't go to the cells that don't express the antigen. We developed systems to look at the condition of binding on their flow, and then finally we took these in animal models and showed that if you put a drug like a taxane drug, like docetaxel, and create xenotransplants of tumors, that an injection, in this case, intratumoral injection, would be highly efficacious for treatment of those cancers. Now, by this time, then I asked, well, could this actually become a real drug? And the feedback that I got from a colleague who was in the industry was probably not. And the reason is that every time you make this nanoparticle using a dye block system that Roxandra had made, but then go to conjugate ligands on the surface, that the chemistry doesn't always work the same. And so ligand density may be different during the conjugation step. Drugs may leak out. So with that, we went back to the drawing board and we created our own system, which was a tri-block system. And the tri-block built on the dye block that Roxandra had made, the PLGMOD hydrophobic entity can encapsulate drug. The pegylated portion would be on the surface of the nanoparticles. But to that, we attached the 57 base pair 2' fluoro-modified RNA moiety that bound to PSMA. And that RNA is hydrophilic. So the system becomes very amphiphilic, a very significant hydrophilic end and a hydrophobic end. But then importantly, if you change the ratio of the new tri-block with the dye block in different mixing ratios, let's say 1:10, 1:20, or 1:40, you could precisely control ligand density on the surface. And when we did that, a really interesting observation happened, which is as you add more and more ligand, this isn't always better in terms of targeting. In fact, the optimal targeting shown there in the bottom in that dark blue is actually a targeted system that is not maximally targeted. But the question is why? If you look at the panel on the upper right, the reason is that as you add more and more ligands on the surface of the particles actually become relatively immunogenic and they get quickly taken up by the liver. And so a maximally targeted particle is actually cleared quickly so it doesn't get to the tumor. So when this happened, then this aha moment went on, which we said, okay, well, if that's the case, let's combinatorially create libraries of particles and tune them for their biophysicochemical properties. And in 2006, we started this company called Bind Therapeutics. And the idea behind Bind was to do exactly that. So Bind then developed a drug called the Bind 014 that was a taxane encapsulated targeted nanoparticle that targeted PSMA and optimize it for various different physicochemical properties. And when you do that, you can have a very long circulating particle shown there in the upper left. That's the pharmacokinetic curve where the polymeric backbone of the particle is labeled. So you're chasing the particle in circulation. And what you can see there is that that particle actually stays in plasma for a long time and doesn't get really taken up by the liver or kidney very quickly. But because these are controlled release polymeric system and the particle has a long circulation time, by tuning the rate of drug release, for example, in the upper right side, you can have a rapidly releasing drug shown there in that blue curve, or a more slow releasing drug, maybe in the purple one. You could now, reverse the rapidly releasing one in purple, and slow releasing one in black. You could now fine tune the pharmacokinetic of the drug, not of the particle, shown there in the upper middle. And when you do that, you can really get high level of drug concentration in the tumor. Now, importantly, that showed also to be the case in human. As you can see, that's the pharmacokinetic studies done in patients, actually in healthy subjects in the bottom right. So the concept then is that you design your nanoparticle, drug is inside, you inject it in the body, the particles goes around the vasculature, and then the ligand then binds to the surface antigens, and then it gets endocytosed. And through this phenomena uh, of EPR, which by the way, we have learned so much since that term was coined, that particles can also escape the vasculature, and you get some intratumoral penetration that extends beyond the vascular permeability, which by the way is characteristics of many tumors. Okay, so then the drug then went into human, and this is one of the patients that ended up getting it. This is Evelyn Sorensen, and Evelyn was a patient with cervical CA, and she was refractory to the therapies that she had received. And when she was dosed with Binder-1/4, in oncology, you can get various different levels of response, complete responses when there is no detectable level of tumor. And so she ended up being a CR, given the degree of tumor reduction that she had. And I was thrilled by that. And she ended up speaking to the press, and we didn't know that. And suddenly the phones were ringing off the hook because everybody with cancer who had some relative wanted Binder-1/4, and the drug and the study got presented at the ACR. It was on the cover of Science Translational, but importantly, the National Cancer Institute initially started funding nanotechnology in 2004. And so this was really the first NCI-funded work to go into human and the first great results to come of it. And so Father has a lot of success. I remember cancer.gov had a picture of that nanoparticle, a gigantic picture of it on their homepage, and Harvard had it on their homepage, and MIT had it on their homepage, and Prostate Cancer Foundation that funded it had it on their homepage. And so that was wonderful. And so with that then, an article got written that said, this is the dawn of nanomedicine, and there's a particle that is kind of rising, and that's the sun. And then what happened was success. Then the company went public, and so everything is terrific. But then phase 2 data came out, and Binder-1/4 was statistically no better than the parent drug docetaxel. And I remember I was presenting the final clinical data, and and someone in the audience was a very prominent cancer biologist in Boston, who I will not name, said to me, Omid, is this the dawn of nanomedicine, or is the sun going down in nanomedicine? Which I think is a very funny comment looking at that particle. And I would actually say it's neither. This is an example where we iterate and we learn, and boy, was there a lot of learning in this. So for example, one of the issues that we had with particles going into human were that they were immunogenic, and I had asked a colleague of mine, Uli Van Andrian, who's a professor at HMS, to come join our SAB, and Uli was helping us against immunogenicity. And Uli said, Omid, you guys design particles and you can combinatorially make them any way you want. But what if you combinatorially made it so that it acts as immune modulator? Because the immune system over millions of years has been designed to respond to, if you would, structures at the nanoscale. Like viruses are in the tens of nanometer typically, and bacteria, hundreds of nanometer or micrometer size. And so that pattern recognition from an immune system is designed for that system. And so you can put immune modulator inside. We can maybe decorate the surface of the nanoparticles with various different things. And therefore you can create systems for immune modulations, and which can be either immune activation or immune tolerance. Man, that was great. And so we started another company together with Uli and also Bob Langer, and that was Selecta. That was a 2008 start. And so Selecta ended up doing exactly that. And the company then went also public, and then it brought a drug into the clinic called SEL-212. And SEL-212 finally finished phase 3 clinical trial in 2023, and met all its endpoints. for treatment of refractory gout. And that drug earlier in 2024, a BLA application was filed, and so that drug is now pending FDA approval. So this is an example where you learn and you iterate. Now, what did we learn in the context of the bind experience? Kind of going back to that for a second. So when you inject particles in a system, very quickly, as Kenneth Dawson very eloquently wrote in his 2007 PNAS paper, a layer of macromolecule formed on the surface. That is what is now referred to as the biological identity of that engineered nanoparticle. Now, if you create particles with various different, for example, just simple density of polyethylene glycol on the surface, just to change surface hydrophilicity, and do that in particles of different sizes. When you do that, and you then inject this in mice, for example, you see that various different ligand density suddenly results in early clearance or not. For example, in that ApoE knockout mice versus healthy mice. But if you look at the LDL receptor knockout mice, response would be different. So different biological system, different kind of a response in terms of clearance. But what is driving that? If you look at, for example, just the density of PEG on the surface, and you inject and then you capture the particles, you release the proteins, and you look at the proteins under mass spec, you begin to see that the protein corona composition of these different systems is different in those biological systems. That is what is meant by the biological identity. Now, this is important because when you're developing nanoparticle systems for in vivo applications like therapeutics, you're limited in your choice of nanomaterials because the nanomaterial needs to be safe in a human system. But if you're developing nanoparticle systems for ex vivo application in a test tube, for example, then your canvas of engineering and design is almost unlimited because you can design any nanomaterial that you want, therefore at equilibrium can have any kind of a protein corona that you want. And so that was another aha moment. For example, between Bind and Selecta, solving the immunogenicity problem resulted in the formation of Selecta, and now on the cusp of an FDA-approved drug. In the context of this problem, we said, well, what if we leverage this protein corona binding to really capture proteins for research applications or maybe clinical applications? And that was important because in the 2 decades before, really kind of going back to 2006, with the launch of next generation sequencing platforms, access to genomic content had exploded. And by the way, cost had come down. The first time we sequenced the human genome, 15 years and $2.3 billion. And as of now, you could do $100 sequencing of a whole human genome. And by the way, one company, Ultima, announced that they'll do it for free for a set of subjects. But so cost of sequencing is coming down, which means that more and more organisms are getting sequenced. But sequence gives you essentially your to a moderate level your status, largely your risk. It's the code, but what is happening to you at that moment, your functional status is really driven by what is the protein content of your body at the time. Like, for example, two genetically identical twins, one can get a disease and one doesn't. Genetically, they're identical, but one ends up with a disease and one doesn't. And why is that? A lot of epigenetic factors, but at the end of the day, their proteomic signature, if you looked at it, would be different. Now, with that said, access to the proteome was limited. Now, this is important because as you go from the left side of this graph to the right side, you go from the genome to the proteome side, complexity escalates. So we all have about 20,000 genes, but we end up with about 200,000 transcripts through different RNA processing. And then from there, you end up million plus multiples of millions of different protein variant. Those variants can be at the level of an isoform. There's about 70,000 protein isoforms that each of us have, which then if you add genetic variant on top of it, so new stop codons and new splice sites that are driven by our SNP profile, then splice forms would be much, much higher than 70,000. On top of that, post-translational modification can change proteins, and proteins work by forming complexes together. So as you can see, the complexity of the proteome is much, much deeper than the complexity of the genome. In fact, if you look at a study published looking at the UK Biobank, where 455,000 exomes were sequenced, that just by sequence alone, and this does not include PTMs or others, that there's an expectation that in that 450,000 subjects, there's about 8.9 million protein variants just by sequence alone. Now, some of those variants may change conformation of the protein and some may not, but there's so much about the proteome that we didn't understand, and that was the real motivation to do this. And the problem was the technologies to get deep into the proteome at scale to do a similar study as at UKBB was just not possible. Why? Because proteins are not only complex in terms of their structure, they're also complex in terms of their quantity. What you can see there in the graph on the upper left, that's the human protein, for example, in plasma. In the upper left are the most abundant protein, and the lower right are the least abundant protein. Albumin alone is about 50% of total protein mass in plasma. Albumin plus the next 21 proteins represent 99% of total protein mass. So what that means is that the remaining thousands of protein that follows are 1% of protein mass. And so to get to that depth, you needed to do really complex processing. And so folks that did this unbiased proteomics, they would do immunodepletion of abundant protein, a series of fractionations to go deeper and deeper into the proteome. And that's because the systems used for that applications did not match the dynamic range complexity of the plasma. Typical mass spec used for unbiased proteomic is 4 to 5 order of magnitude in terms of its capability to detect, whereas the human plasma can be 10 order of magnitude in terms of its between the most abundant and least abundant proteins. Okay. So that was the motivation then to start Seer, to really decode the biology of the proteome. So I'm now going to shift from an academic work to the work that Seer is doing. And the idea was to really democratize access to the proteome. So this is the history of Seer. I mean, a lot of the science almost dates back to 2008. I mentioned that Professor Dawson had published a 2007 paper of protein corona. We were working on the same space, for example, looking at precise engineering of particles with different surfaces. We had published another paper that showed that different surfaces can activate the immune system in a different way. But the recognition that physicochemical properties drives the very specific signature of the proteins. We had published another paper in 2017 that I showed you that looked at protein corona in vivo. That was actually the first example of such a study. So we started a company. It got incorporated in 2017. We did a series of fundraising, and then COVID dawned upon all of us in 2020. And the team really persevered. We viewed what we were doing as important to health and to science. And so during COVID, the team continued to work. We tried to work in a distributed way so that people weren't close to each other. Teams were working in shifts, but they kept plowing through it. We shipped our first instrument in December of 2020, and by that time, Seer went public. So today, Seer is in this proteomic space, which is also but taps into the genomic space because the real future is the integration of this omics. We've detected over 36,000 protein across different species, served over 120 customers globally in 20 different countries. The company is well capitalized. We also spun out a company called Prognomic that we own 20% of. It's a diagnostic company looking at early detection of lung cancer using proteomic signatures. Now, if you look at the proteome as that iceberg, if you approach proteome in a targeted way, meaning I'm going to go look at it with an antibody in a targeted way and detect the protein. When you do that, though, you're only seeing what you're intended to see, but a lot of the lion's share of in the proteome is actually what's submerged under the water that you don't know. And so looking at the proteome in an unbiased way is important for discovery application. And as I said, the challenge was in the upstream workflow that did not scale. And so we developed a system, and I'll get into the weeds in terms of what it does and how it works in a couple of minutes. But we solved that problem by releasing an instrument called the SP100 and then an assay initially called the RISE, and subsequently we upgraded the assay to what we call the XT assay. So by this time, we had enabled large-scale, deep, unbiased proteomic studies to be done. And long and behold, we created a new bottleneck for ourselves. A new bottleneck was data analysis, because this vast amount of proteomic data was never possible to create. And so a way to analyze it was never solved because that was never a problem that existed. So we then created systems for analysis of that large-scale proteomic data, launched the Proteograph Analysis Suite, and then the 2.0 and then the 3.0 version of that. And today, customers can now analyze proteins using a mass spec and the proteograph in ways that was not possible before. The company now has its own large patent portfolio and independent of the work that I've done as an academic. And it's important to just look at how the trajectory of proteomics changed really just in the last 4 years. The deepest study ever published in plasma proteome was a study from Broad Institute from Steve Carr's group. And that looked at 5,300 proteins in about 16 samples. Where are we today? By the way, Broad has one of our instruments, but where are we today? Today, customers can get to 13,000 proteins across their studies. And these customers are beginning to publish their own papers. About 30 papers has been published across these 120 customers. And they're now doing it at scale. We saw population scale proteomic studies get done using targeted approaches. And I had predicted about a year ago that the first population scale deep unbiased proteomics will be done in 2025. And I'm optimistic that that would be the case. So hopefully, as you follow the field, that you'll see those things will be forthcoming. Something that was just not possible when the largest study was 16 samples, and now studies are being done at the population level. The magnitude of changing the arc of proteomic is quite dramatic. So how deep can you go with Seer? So if you take a typical mass spec and just do a direct injection of a digested protein in it, with the latest and the greatest of technologies, which is the Thermo Fisher Orbitrap Astral mass spectrometers, you can get to about maybe just about 770 proteins. But if you first put the sample through the Proteograph, it's an 8-hour automated workflow, and it runs 40 samples at once. You can get to 6,000 proteins. If you do this in a study of scale, like for example, our customer did this across 2,800 subjects, human subjects, because of biological variability, you can get to about 13,000 proteins. And importantly, this improvement in content is not a function of an improvement in the mass spec, because mass specs are getting better and better. What you can see there in the dark blue are different generations of the mass specs over the last few years. And you can see that they're getting better and better. But no matter how much better they are, and you can see that dark blue box keeps growing, when you put a Proteograph upstream to it, the magnitude of the improvement is still quite dramatic. And so I'm proud that in a short 3.5, 4 years since the product was launched, we've shifted from being really a pioneer in this space uh, to now a trusted partner in this space with our customers who are now continuing to publish their own papers and pushing the envelope in their own science. And it's really thrilling for me as an academic to watch access to the proteome become more and more democratized. But how does the system actually work? So I mentioned that Professor Dawson had shown and opened up the field of protein corona for the rest of us. And when you create libraries of nanoparticles that differ from each other in their physicochemical properties, these particles can then interrogate um, the plasma proteome in a unique way so that, for example, they will form different protein coronas. You can then, with that mindset, assemble panels of particles that instead of using a very complicated workflow for fractionation depletion, to really put that entire labor, if you would, on the surface of a nanoparticle in automated workflow and do deep unbiased proteomic at scale. Now, in order for something to actually be functional, it's okay for it to be theoretically working or even experimentally shown to work. But if you want to make it work in a way that it's scalable, it has to be robust. If I do a study today in Boston and someone does a similar study in Copenhagen in Denmark, the studies have to match. If I do a study today and it's a long study that takes a year to finish, and so I go through different batches of consumable reagents, the study needs to be robust. And so accuracy and precision is key. One way to show that is by mixing different species plasma, for example, bovine and human at different ratios, to show that the mass spec, as you're mixing different ratios, the mass spec follows that, and that the expected ratio is also the measured ratio. So when you do that, for example, if you do a neat digest of different ratios of bovine and human, and that's is shown there in the gray, if it falls on that dashed line, then that means in different ratios of mixing, the mass spec is doing a good job. As you can see, when you do this with the proteograph, matching the same proteins that you see with the neat digest, so that's about 10% of the proteins that you would see, or 15% of protein that you would see, or that all the protein that it sees, is you mix it at different ratios that you consistently see that. So the accuracy of the system is quite high. So what are customers doing with this? So here's a collaboration with folks at Mass General, looking at 1,800 samples that they had collected across 10 years, looking at Alzheimer's disease or cognitive decline in subjects and comparing it to folks that didn't have a cognitive decline, otherwise healthy subject, and to see, is there a difference in their proteome during that process of cognitive decline over that decade? There's 138 proteins that actually goes up or down during that decade that correlates to the up or down regulation of their cognitive function. Importantly, only 34 of them in the literature have been known to be associated with AD. 94 of them are putative new biomarkers of a disease that needs to be investigated. If you looked at this in a targeted way, you would miss 55% of them because antibodies are not made for them to be on a targeted panel. Now, MGH then asked a really interesting question, which was, is there a subset of that 138 proteins that if you had them, your velocity of cognitive decline would be quite dramatic? And they identified 8, by the way, 75% of which are not on these targeted panels, that if you have, the velocity of cognitive decline would be rapid. And so that's a clinical score that can be helpful to them. I mentioned that we spun out a company that is in the diagnostic space called Prognomic. Now, Prognomic is an independent company. It's in the Bay Area. I have the pleasure of serving on the board. and has a fantastic team. And they did the largest deep multi-omic study ever attempted. They looked at various different omes, transcriptome, genome, copy number variant, deep unbiased proteome, targeted proteome, metabolome, lipidome. And when they looked at that and they began to see what is driving, is there a signal that can help them detect early stage lung cancer versus healthy? And by early stage, I mean stage 1 versus healthy. And what they have now developed is a clinical test that will likely launch this quarter in Q2 of 2025. And that test will help them distinguish early detection of lung versus healthy subject. And what's driving that classification, you can see there in that receiver operator curve or ROC curve on the left side, largely is the proteomic signature. And importantly, as you can see there on the right, that proteome signature is across the entire dynamic range. Some of them are the proteins that are deep, meaning low abundant protein, but some high abundant proteins. By the way, I mentioned that the proteome is complex because we have different isoforms of proteins. Our genes, through splice variant, can create different proteins. Like, for example, if you look at the top, the upper right side, if you look at proteins using an antibody at the protein level, so those are two different splice variants from the same gene. If the antibody is binding to that orange portion, that antibody can distinguish splice variant 1 from 2. Conversely, if the antibody bound to the gray portion, not shown on the slide, but if it did, it would only pick up splice variant 1, and it wouldn't even know that splice variant 2 was present. But if you looked at this at a peptide level, you could actually distinguish splice variant 1 and 2 from each other. So here's a proof point. If you look at early stage lung and you ask a question, is there a difference between bone morphogenic protein 1, BMP1, which is largely understood to be a housekeeping protein, between lung cancer and healthy? If you looked at this at the protein level, you would reach a conclusion that there is no difference between healthy and lung cancer in BMP1. But if you look at it at the peptide level, BMP1 exists in 4 different isoforms, a short form and 3 long form. And we can see there that the short form is actually upregulated during cancer progression. Um, and versus the long form is actually more present in the healthy subjects. You would have missed that if you had done this. Here's another one. If you now have access to the genome, which thanks to our colleagues for the last 20 years in the genomic space, really crediting the company Illumina and other scientists at Illumina who really enabled it, we do have access to the genome. If you now sequence people, and then you also in parallel do their proteomic analysis, with knowing their genetic differences and their genetic variant, you can now pick up protein variant in those same subjects. For example, this particular subject is at this particular loci has one copy from mom, one from his dad, and has 2 different variants of the protein. One variant that comes from one parent and another variant that comes from another parent. And so I really think that by impedance matching, access to unbiased genome, which we've now had, and by the way, cost has plummeted, and access to the proteome, and I hope we can make the same reduction in cost. The system, we've been able to decrease our cost considerably since launch, and I'm optimistic that it'll continue to go down. Really, the field of proteogenomics is going to enable an astronomical amount of discovery when you match genomic content to proteomic content at scale. So with that, then I want to thank my team at Seer, who really enabled all the second half of the slides that I showed you. And again, I have the pleasure of sharing this story with you, but this is the team that actually did the work and brought it from ideas into practice and into reality of life. So with that, I'm open to questions.
Thank you so much for the amazing speech, Dr. Farokhzad. We're so lucky to have you. I think we're going to be facilitating some Q&A. So I'll just let those trickle in for a second. But then I know also among our team, we have plenty of questions that we would love to ask as well. So I'll just take a few seconds and then we'll start facilitating Q&A. I think, Dr. Farokhzad, I think we're going to ask a few questions first, and then we're going to go to the chat. So I'm just going to ask from our side. So one of my questions was, you've taken several companies public throughout your career, as you've discussed, and I was just wondering if you could share more with us about the process from taking a drug or company from like the R&D phase and then all the way to an initial public offering into the public market. I think you clearly have a lot of experience with that, so hearing more about that would be great.
And then Julia, just a question for you. Are you interested in learning even earlier, how does a company actually even get formed? Or are we assuming that the company exists and you're curious of the process to take it public?
Honestly, I think hearing about starting from the preliminary lab phase, so
Okay.
before a company, maybe preliminary research would be awesome to hear.
So Julia, I think the most important thing is that that you got to protect your ideas, and so you have to file patents. And the reason is that if you don't have patents, then it's impossible to get investor interest. Risk capital comes in when, and I don't mean this to be mean, but when those investors know that you can protect your ideas and you can block others from replicating what you've done. So the most important thing then is that as you're doing experiments and discovering new things, before you disclose any of it publicly, you file patents, and the universities have become very, very smart at encouraging you in doing that. Now, as an academic, you're an inventor, and inventorship is really a matter of law. But the owner of the patent is the university. And so once you file your patents and there's a number of discoveries that you've made, there's a time that you need to make a decision of how do you commercialize that. And one option, which is the option I've taken, it's not necessarily the only option, but is that you actually approach the university and say, look, you are the owner of the IP. Can I license it from you? And so you actually become the entity that ends up licensing the technology from the university. So you form your own company. That's an easy thing to do, a couple $100 and a good lawyer. They incorporate your entity, and then the entity goes and license the IP from the university. By the way, no different than if Pfizer or Merck or any other company went to license it. The university has some expectation in terms of what it wants to get financially for the IP. They will ask for you to be responsible for that. So then you license the IP into this entity. And with that, then you go out and you ask for investors. Now there's investors come in different flavors. A lot of the investors who are in this early stage life science space know the risk that it takes to take something from an early academic work forward. If they feel that their work has been de-risked appropriately in an academic lab, then you get some initial capital. That initial capital tends to be the most expensive capital you ever raise because the proof points are relatively scarce at that point. So you raise some capital. The way I did it for the initial, I would say 15 years of my life when I was an academic was that I would then try to collaborate with industry colleagues and build a team, just like you build a team in your academic lab and you hire amazing postdocs and students who do all the wonderful work in an academic lab. You then go out and you recruit a different set of talent, which is folks that are in the industry that come, they still believe in your vision and your mission, and they come and they become your partners and your colleagues in your company. And you could then choose various different ways to contribute. At that point, you can be on their scientific advisory board, you can be on the board of directors. So you form an entity, and this entity raises capital, and you begin to first replicate some of the work that was an academic to make sure it's actually valid. And then the degree of robustness that happens in the industry is actually very, very high, much, much higher than in an academic lab. Things like chemistry, manufacturing, and control were terms I had never even heard of, which is what the FDA requires in order to allow something to go into human. You need to have very high level of rigor in those principles. And you do a lot of those studies. You then move into different types of animal models. By the way, I don't know if you saw, just a few days ago, FDA then said that they're going to get away from animal models altogether with looking at model systems and including data systems, which I think is a wonderful thing for the industry. And then you raise more capital, you go into human. The initial clinical trials are phase one. They're about safety. They tend to be healthy subjects, usually, not always. And you dose escalate to understand the tolerability of your system. And then the rubber hits the road when you enter phase two, because by then you're looking at efficacy. And then you do phase two, which are larger, much larger studies. You tend to replicate them again. And then you go into phase three and then finally approval. Important thing is that when you go into the clinic, about 6% of the drugs that enter clinical trial end up being approved at the back end. And importantly, of the drugs that are approved, actually a relatively small fraction of them will have a positive return on investment. So the way the field really works is it works because it's looking for blockbusters. If you have a relatively small drug in terms of its market share, it doesn't return the investment that one makes. Now, the dynamic around drugs are changing because of cell and gene therapy drugs sometimes can cost millions per treatment. So those are small drugs, but they're being charged a lot to kind of recoup the R&D investments that's being made. But and also, Julia, it's learning no different than learning science in university or grad school, or the process of building companies is a whole new learning experience, and I think it's wonderful and really, really rewarding, actually.
Thank you so much. And then that was an amazing answer. And we have a bunch of questions in the Q&A chat. And to paraphrase one, so I know you were a physician before and you were a professor at Harvard Medical School. So what was it like for you to make the transition from academia and being a physician to like biotech and the public sector, I guess. And I think there's a lot of risk involved in that too. So maybe you could talk, I know you touched on this a little bit in the last question, but it would be great to hear about that decision that you made and what your motivators were.
So I would say that, I was fortunate enough that I trained in the laboratory of Professor Langer, who is obviously probably the Edison of our time and the most decorated scientist, but also an incredibly prolific entrepreneur. And so the process of starting companies was something that I would see around me when I was training always. And in fact, many of Bob's former students and postdocs have gone on to start companies. But if I look at other colleagues and if I look at my own experience, you gotta have conviction about what you're doing, because in science as an academic, everything we do is sales. And by that I mean you write your grant, that's a sales pitch, to the NIH, and then some reviewer reviews your grant, and they may fund it or they may bash it. And if they bash it, you need to go back to them and to try to convince them again that it's actually a really, really worthwhile science. And so that's, you're repeating your sales pitch, trying to overcome rebuttals. Same exact thing that happens with an investor. You pitch them, they'll be critical. You try to refine your pitch, you come back. Key is always, probably the most important criteria I would say is that you need A++ people in an academic lab. You need A++ people in an industry setting to move things forward. If you are true to what you know and you're open to learn, just like you are as an academic, and you approach it that way in an industry setting, then I think you'll be very successful. For me, the transition was relatively seamless. I was 2 years into my faculty job when I started my first company. And while I was running an academic lab, I served on boards. And so by the time I actually left, Seer was the fifth company I had started, and it was the third public company board that I had served, and the second that I had chaired. So I felt that the transition was relatively smooth. It wasn't that I was a fish out of the water. I had experienced it over a long amount of time. Um, and by the way, Julia, at some point, I probably go back to the academic world again. So the path is bi-directional. It's very fluid. Um, and you could start off as an academic, go into industry and come back, or be in the industry and later go into academic. And so it's been great. And I would actually encourage folks to really not consider them vastly different. To me, there's a lot of similarities, actually.
Thank you so much. We have a question from Kenzo in the chat. He said, thank you so much for your intellectually stimulating keynote on a nanoparticle technology and proteomics innovation. I'm genuinely inspired on your journey from HMS to leading Seer. Given the impressive scalability and depth of your proteographic XT platform achieves, what specific clinical applications do you believe will first benefit meaningfully from this technology? Additionally, what unique regulatory challenges do you anticipate in translating this nanoparticle-based proteomics approach into validated clinical diagnostics that can demonstrate clear utility to both the FDA and healthcare stakeholders?
Well, a fantastic question. I appreciate that. So let me tackle it maybe in reverse, which is the product is an RUO product. It means research use only. So it is not meant for clinical applications. If you look at NGS, next generation sequencing, that got launched, that product was RUO for many, many years before it got cleared for clinical use. Now, and so my expectation is that eventually it'll get into clinical use, but today is an RUO product. So now let me go back to the first part of the question, which was, what are the applications for proteomics, actually, I would say, in general? I would say anything that you can think of that genomic touched, I expect proteomics will touch. In addition, I think there are areas that genomic touched but wasn't helpful that I think proteomic will have a shot at being helpful. So biomarker discovery, target validation, diagnostics, ag bio, all of those are fair game to be explored for the proteomics. But for example, there was not really interesting genomic signature that emerged for neurodegenerative diseases, but I expect so genomic didn't make a real impact there. But I do think that, for example, proteomics will make an impact in the neurodegenerative diseases. So I think that if you look at what NGS enabled, and that was largely driven by one company, Illumina, the end market that it created, and then those end markets expanded, one couldn't even imagine before. So areas like minimal residual disease after surgical resection, for example, looking at recurrence is something that genomic testing now is huge. In fact, the companies that are now marketing those products are larger in scale and their market cap than Illumina, who actually invented the platform for sequencing them. Or technologies for early detection, like company Grail, and by the way, there's a number of them, Guardant Health, Exact, Prognomic, Freenome, many examples that are doing those. So I think proteomic is going to make an impact in virtually every area that genomic did and expand those end markets, plus create entirely new end markets that don't even exist today, some that I can't probably even imagine today, and that we'll see what happens in the next 10 and 20 years. But we are literally at the very, very beginning of what I call the proteomics revolution. Our understanding of the genome has exploded. We are just at the beginning of content revelation in terms of proteomic. And so that content is going to be forthcoming for years to come, and an enormous amount of insights going to be gained in that process.
Amazing. And I think this might be our last question. But just to wrap up our discussion, I think one pressing question, most of us are undergraduates on this Zoom. And so we would love to hear what your advice would be to an undergraduate student interested in pursuing a career in biomedical research and biotechnology. What are the opportunities or challenges that we might face, and also now in like a new era with AI research, what do you think are the skills that we should be focused on learning and building as we go through our undergraduate career, maybe as we're working in labs, etc.?
Okay. Julia, I think the most important thing is that in life, and this is not just during your undergraduate phase, but in every aspect of your life, you need to have great partners and great mentors that really care about you. And ideally, these are folks who have walked the path that you want to walk before successfully. And so you're stepping in their footsteps, and that they constantly want to lift you up. So I would say spend time in figuring out who those people are and gravitate toward them. So that's important message one. I think you'll go so much further by people pushing you forward than if you have headwind that's pulling you back. Okay. Next is follow your passion, never follow a fad or a trend. Because scientific fads and trends will come and go. And the half-life is much, much shorter than your career. So if you try to chase a trend, by the time you become an expert at it, there is another trend. And then there's another trend. And you're always chasing the next trend. So find out where lies your deep curiosity and become the deepest person in that space. Instead of being the broadest person and understanding a lot at a shallow level, try to be the deepest person in what you're really, really passionate about. And in that context, it almost doesn't matter in terms of the very specific area, because if you're deep, odds are high that you will make a massive contribution in that space. Okay, now, so those are the two principles that I think will hold no matter what the rest of what I say is. But here's the rest of it, which is we've made enormous advances in understanding diseases like cancer and cardiovascular. And of course, there's a ton of room in that space to continue to make improvements. But our understanding of the human brain is at its infancy. And so I think the next decades are going to be the decades of really understanding how the total system operates. And so understanding these neuronal connections, their relationship with inflammation and just about everything, the process of memory formation and neurodegenerative diseases. I mean, the economic tax of an aging population and the neurodegenerative diseases that would be forthcoming with that is massive. So I think it's an exciting time to be in the neurobiology and neurotechnology space. And it's also the era of omes and omics and the massive amount of data that comes with that. AI and understanding that data is going to be key. Once we have proteomic content at the population scale across various different health and disease status, that massive amount of data needs to be understood. So always try to look at a problem, and this is what I say to my own kids, and Julia, you know, Ryan and Nika, is that try to approach the problem in an orthogonal way. Train yourself in two totally independent fields so that your lens is a different lens than just being myopic in terms of one view. So being extremely good, for example, at neurobiology, neurotechnology, but really coming at it in terms of data is very, very powerful. So, so I would say expose yourself to a lot of different areas to understand where you're passionate about, and then go deep in your areas of passion, and don't shy away from disciplines that you're uncomfortable with, that you don't understand, because that is really where innovation actually happens. It's that cross section of disciplines where new light is shined in problems that are otherwise very, very difficult to understand.
That was a fantastic response. I think we can all step away from this conversation with lots of fantastic advice. Thank you so much again for coming to speak with us and for facilitating that Q&A. To everyone who asked questions that we didn't get to, we're so sorry. But thank you for asking them anyway. And yeah, Dr. Farokhzad, thank you so, so much
Thank you
for
to you
coming.
and your team for inviting me. Have a fantastic
Of
rest of
course.
the
We are
conference.
thrilled to have you.
Okay, take good care, everyone. Bye-bye.
You too. Bye, Dr. Farokhzad.
So at this time, we'll be transitioning to the workshop portion of this conference. Workshops are going to be held by Cornell iGEM, NYU iGEM, Stanford Biome, the Harvard Undergraduate Biotechnology Club, and the Harvard Undergraduate OpenBio Laboratory team. We have 5 different workshops going on, and I'm going to put the link in the chat right now that tells you descriptions of each of the workshops so you can decide which one to join. As a reminder of the schedule for the day, workshops will be occurring from 10:30 to 11:30. And then from 11:30 to 12, we're going to take a lunch break. Please be back on this main Zoom by 12:15 because we have our next speaker, Dr. Ed Boyden. So I'm putting that link in the chat right now. This document has all the information on the workshops that you should be attending in the next hour. So pick one that seems the most interesting to you and please stay there until 11:30. And we'll see you back on this link, which is at the top of that document at 12:15.
Thank you guys.
Hello.
Hello. Hi, Dr. Boyden.
Hey, how are you doing?
Good. How are you?
Doing all right. Doing all right.
Welcome back to all our attendees. We're going to get started with our second keynote speaker session. It's my absolute honor to introduce our keynote speaker, Dr. Ed Boyden. Dr. Boyden is the Eva Tan Professor in Neurotechnology at MIT and an investigator at the McGovern Institute for Brain Research. He's internationally recognized for pioneering powerful tools that help us understand and even reprogram the brain. He co-invented optogenetics, a revolutionary technique that uses light to control neurons, and has since led innovations in brain mapping, expansion microscopy, and neural recording technologies. His work sits at the intersection of neuroscience, engineering, and biotechnology, and it's already transforming how we study and treat neurological disorders. Dr. Boyden's numerous honors include the Breakthrough Prize in Life Sciences, the Canada Gardner International Award, the BBVA Foundation Frontiers of Knowledge Award, the Wilhelm Exner Medal, and the election to National Academies of Sciences and the American Academy of Arts and Sciences. So please join me in welcoming Dr. Ed Boyden.
Thank you so much for the invitation. It's great to be here, even virtually. I'll screen share in a second, but yeah, I direct a group at MIT that works on technologies for imaging and controlling biological systems. And my ultimate hope is that we can completely understand biology. And I guess the way I think about it is scientific fields have this moment of what I like to call the ground truth. In physics, that was quantum mechanics, right? So now you know the particles and how they interact. And as far as, much of physics on the surface of the Earth, it's a pretty good ground truth. And so after quantum mechanics, we got microchips and lasers and the internet and cell phones, and the list goes on and on and on. Now, chemistry hit its ground truth. The periodic table, that's the parts, and then molecular bonds, right? So again, if you have the list of parts and how they work together, now you can make polymers and all sorts of things, right? Metamaterials, and the list goes on and on. So why is biology special? I mean, let's find all the parts and how they work together. Now, in some ways, biology is special. There are too many kinds of parts. So in physics, you have a couple particles. In chemistry, you have, let's say, a typical chemist might deal with one or two dozen elements and kinds of bonds and so forth, right? But in biology, there are hundreds of thousands to millions to who knows how many things. And so we need to take that into account. So today I want to tell you a couple short stories about tools that we built to really try to take into account the fundamental heterogeneity of the building blocks of biological systems. My dream is we can understand a biological system, and probably because there's so many parts, that means making computer models, right? In physics, there's a small number of parts, chemistry, a medium number, biology, a colossal number. So we need to be able to simulate it in the computer. Now, how do we get the list of the parts and how they work together? Well, I'll tell you about 3 tool sets we've been building that allow us to map molecules and what they do, control dynamics of systems, and observe dynamics of systems. And in all 3 cases, part of the challenge is dealing with the heterogeneity of biological systems. But we also have to think about the scale, spatial scale and temporal scale. So for the spatial scale, biomolecules are tiny, but the body is large. So we need to be able to map molecules across the body. I'll tell you about expansion microscopy, which is our invention that lets you do this. Then we have the time dimension. How can you see and control things over time? And for that, I'll tell you about optogenetics, where we control brain activity with light, and then tools for imaging high-speed signals in biological systems. So to summarize, there are really 3 axes along which biology is very challenging compared to earlier sciences, such as chemistry and physics. Heterogeneity, too many parts, spatial scale from atomic to entire body, and temporal scale from millisecond or even briefer to lifespan. Let's start by talking about space. How can you image something with nanoscale precision? Well, many people have invented technologies to do that. Super resolution microscopes, for example, electron microscopes, but all of them struggle to deal with large 3D objects. How can you image a large object with nano precision? Well, in our group, we often think about being the opposite of what people have done before. And so we thought for 300 years, the way you image is you magnify the image with some kind of lensing effect, right? You magnify the energy or the information transmitted to, from, or through an object. And so we thought, what if we just make the darn thing bigger? Let's take a biological specimen, not living, and form a dense mesh of swellable polymer throughout it. Basically the stuff you find in baby diapers. If you do it evenly enough, inside cells and outside cells, in between biomolecules and around biomolecules, then we thought if we add water, the baby diaper material, sodium polyacrylate, will swell, and then we can get a physical expansion of the biological system. So we first announced this discovery in 2015. Here in panel B, this is work done by 2 then students in the lab, Faith Chen and Paul Tilbert. In panel B is a piece of the mouse brain, a few millimeters on a side. Panel C, the same piece of mouse brain, about a day and a half later. We've blown it up 100 times in volume, about 4 times in each direction. And the polymer thread starts out very dense, like in the upper left. The spacing is around 1 to 2 nanometers, and it ends up like in the lower left, extended. Here's a little movie, half an hour sped up to half a minute, showing a piece of brain expanding. We formed the polymer earlier, and now we're going to add water about there. And so I hope you can see that this piece of brain tissue is growing. So I'll tell you how it works, but before we do that, let me get it out of the way, the frequently asked questions. Sometimes people ask these questions at the end of the lecture, and by then it's kind of too late. So let me get them out of the way now. Sometimes people say, have you injured the cell? Is it unhealthy? It's not alive at all. This does not work on a living thing. So let's get that out of the way. Sometimes people ask, have you disrupted the molecular interactions? Absolutely. We're pulling them all apart from each other. So what's left? Well, the information is left, right? The information that's encoded by the relative organization of biomolecules. And that's what most people care about. The following cartoons illustrate how it works. In the first step of expansion microscopy, we add an anchoring molecule. So in brown are proteins, and in purple, we are little anchors or handles. And we have anchors that can bind to DNA and RNA and all sorts of biomolecules. Next, we form a dense spider web-like mesh of this baby diaper polymer, sodium polyacrylate. The white spheres are sodium acrylate, and they self-assemble into polymer chains, as you see here, through a process called polymerization. And when a polymer chain grows and encounters a handle, it'll form a bond. Next, we have to soften the specimen. We add detergent or heat or water, detergent, heat, or enzymes, and it loosens stuff up. And then when we add water, the sodium polyacrylate will swell as osmotic forces bring in the water. But because of the softening, because of the polymer, and because of those anchors, we get an isotropic expansion. We designed the process to be isotropic, but of course we have to show it. So we took pre-expansion images on a regular super resolution microscope, and then post-expansion images using a conventional microscope. In light microscopy, we often look at fluorescent labels attached to molecules. So here we're using fluorescent antibodies against tubulin. And the purple is pre-expansion, and the green is post-expansion. So the distortion is not zero, but it's really small. That's distortion without resolution. So we know the ground truth of many things from previous super resolution or electron microscopy studies. We can take our data that we see and compare to the known ground truth. So here are microtubules. We know how wide they should be. And so we can estimate the resolution as about 70 nanometers. Now, a typical good microscope would have a resolution of 300 nanometers. 300 divided by 4.5, which is the expansion factor, would give you about 70. And so you might wonder, if you expand more, can you get more resolution, better resolution? And the answer is yes, but there's a lot of things you can do with 4.5-fold expansion that are difficult to do with older techniques. So here is an example mapping the wiring of the brain. This is the mouse hippocampus. Neurons are expressing fluorescent proteins virally delivered. The top middle shows the wiring of the brain on a conventional microscope. It's hard to see, right? It looks fuzzy. The upper right is the same specimen, same microscope as the upper middle, but after expansion. And now you can cleanly resolve the wiring of the brain. That was all protein visualization. What about nucleic acids? Oswasi joined the team, and Yi Kui and Gaoji Yang continued the work. We showed we could anchor and expand nucleic acids and proteins. So here in green is the yellow fluorescent protein, and in pink, we're doing post-expansion in situ hybridization to visualize individual messenger RNAs. So we can now look at two kinds of molecule, right? Nucleic acids and proteins. That's all about fourfold expansion. Can you expand more? Well, when J.B. Chang was in the group, and later Deblina Sarkar, Oswasi, and Jinyoung Kang made a very easy-to-use version of this, we invented the following. Take a specimen, polymerize it and expand it, like I told you before, form a second polymer in the space opened by the first expansion, and expand it again. So 4 times 4 would give you about 20-fold expansion. We realized something else. Expansion not only democratizes nanoimaging, there's another feature. By pulling molecules apart, you can get better labeling. So in the top row of this cartoon, the red and green antibodies can decorate proteins on the outside of a complex, but they can't get inside. If you expand proteins away from each other, though, like in the second row of this cartoon, now the yellow and blue antibodies can make their way in. We thought the way proteins work is often by touching each other. If we expand them away from each other, can we see them better? Here, in panels B, C, and D, we're comparing pre-expansion labeling with post-expansion labeling. Yellow is pre, and pink is post. There's not much yellow to be seen. There's a lot more pink. These are crowded proteins that we've decrowded from each other. Now in panels F, G, and H, you can see that the yellow and the pink are more similar. Not all proteins are crowded. Working with Tom Blampied at the University of Maryland and Li Wei Sai at MIT, we decided to investigate different proteins and see if we could see their organization at synapses. So here are some of the previously invisible proteins. This calcium channel, CAV 2.1, now it can be visualized with post-expansion staining. And to make a long story short, we can see the organization of synapses. The pre- and postsynaptic proteins are coordinated with each other with nanoscale precision. Jinyun Kang and Margaret Schroeder showed that we can also stain, image, and wash out the stains over many cycles of staining and imaging. And then you can align those images with each other, as you can see here at the bottom using computer vision. And now we can get many, many, many visualized proteins in the same specimen. Here we're visualizing 20 different proteins in the same piece of the mouse brain. Now that was 20-fold expansion by iterating it, but lately we also found we could do it in a single step by controlling the oxygen better. In work with Laura Kiesling's group in MIT Chemistry, led by Taishin and Xue Wang, working across our two groups. Pablo Valdez wondered if we can make invisible proteins visible, what about invisible cells? In brain cancers, there are aggressive tumor cells, and they have two prominently expressed proteins, vimentin, shown in cyan, and GFAP, shown in pink. So here, the top three rows are pre-expansion staining. These are actual brain tumor specimens from human patients. The bottom two rows are the same specimen, same field of view, same antibody, same microscope, but with the antibodies added a second time after expansion. So we can see the aggressive tumor cells much more clearly. In fact, if you look where my mouse is pointing, if we counted the aggressive tumor cells, there were 6 times more of them after expansion, when labeled after expansion, than before. Now that's all in the brain, but we can look at other tissues too. When Yongshin Zhao and Octavia Bricure were working with me and many groups at Harvard Medical School, Astrid Wines, Stu Schnitt, Andy Beck, and others, we found that we can apply it to many kinds of human tissues. On the left are normal, and on the right are cancer-containing specimens of prostate, lung, breast, pancreas, and the list goes on and on. It's been 10 years since expansion came out. Probably about almost 800 discoveries or experiments have been published in preprints or in peer-reviewed papers. Our website, expansionmicroscopy.org, has recipes for how to do it. Nature did a little 10-year anniversary news report about it, and it showed some of the really surprising and exciting applications people are doing with it. But one of my favorite quotes was from Yongshin Zhao, now running his own group at CMU. It's so easy, even my 9-year-old can do it. And we have posted on our website photographic step-by-step tutorials so that anybody can learn. And it's been very useful. So to summarize this first part of the talk, we've discovered we can physically magnify objects. It democratizes nanoimaging. It's easy to do. And by decrowding molecules from each other, we make room for better labeling. Probably thousands of groups are doing it by now. And it's not only easy to use, it's easy to improve. Lots of people are adopting it for the microbiome, seeds of plants, you name it. Now let's switch gears. The limitation of expansion, of course, is that it probably will never work on a living thing, right? You're pulling everything apart. So in the last half of the talk, I want to talk about the imaging and control of living things. Let's start with optogenetics. Optogenetics started when Carl Deisroth and I were both students at Stanford University, and we wanted to control the electrical activity of the brain. How can you do that as precisely as possible? Well, we went through basically all the laws of physics, right? There are only so many kinds of energy you can deliver to the brain. Electricity, magnetic fields, ultrasound, light, really a handful of things, right? And we thought light could be the most precise, and of course it's faster than anything else. If only we could figure out how to do it. Just as you bring electrodes into the brain, even of awake, human patients, right? There aren't pain sensors in the brain. Maybe we could bring in optical fibers. Then the question became, how do we make brain cells sense light? I was fascinated by microbial rhodopsins. These are light-driven 7-transmembrane proteins that are in the membranes of single-celled microbes. They look like the protein in the lower left, a 7-transmembrane protein. It has a vitamin A variant called all-transretinal in it. And this sits in the membranes of single-celled microbes and helps them convert sunlight into chemical energy, in particular a proton gradient. The proton gradient then powers the production of ATP. This was discovered half a century ago. People then discovered light-driven chloride pumps. And then around the turn of the century, people found light-driven ion channels. So being ion channels, they don't store energy, but they can be used by single-celled algae to navigate around. Flash forward to the current day. We've found that members of all of these classes can be discovered that are safe enough, fast enough, and powerful enough that they can mediate the optical control of neural activity. So on the left-hand side, when Brian Chow and Shu Han worked with me, we showed that we could express certain light-driven proton pumps in neurons in the awake behaving mouse brain, as you see here, shine colored light, in this case green or yellow light, and we could pump protons out of neurons and turn them off. So by silencing neurons, you can find out what they're needed for. Are they needed for a behavior, or a disease state, or a memory, or an emotion? In the middle column, Shu and later Amy Chuang found light-driven chloride pumps that we could use to silence neural activity with light. So this also has sort of similar and related effects. And on the right-hand side, Carl Deisroth and I originally put light-driven ion channels into brain cells. We showed that blue light could activate them. If you activate a brain cell or cells, you could figure out what it is sufficient to trigger. Can it drive a behavior, or a memory, or an emotion? And Nathan Klepetke and others worked to make this a more perfected toolbox. So just as expansion microscopy has been used by thousands of groups probably by this point, optogenetics, as we call it, opto for light, and genetics because these are genetically encoded reagents, has also been used by thousands of people, mostly in animal models to study disease states or behaviors. But a couple summers ago, a group in Europe took one of our genes, a red light activated cation channel that we showed in a paper in 2014, and they put it into the human eye. Millions of people are blind. They've lost the photoreceptor cells in their eyes. But what if you could take our molecule and put it into the spared cells of the eye? Could you make the eye into a camera? And they showed that would work. It's not perfect vision, but this person could discriminate between different household objects. He could see doors on a hallway. He could see lines of a crosswalk. He had trouble reading and recognizing faces, but still a pretty good partial restoration of functional vision. So I just wanted to go through optogenetics quickly because it's a fairly mature field, but suffice it to say that it was driven by serendipity. We were very lucky. These are molecules from the natural world, and they just worked. We found molecules that were safe enough, fast enough, and strong enough to mediate the optical control of neural electrical activity. Now I want to talk about some new stuff. And here we were not so lucky in the sense that the natural world didn't just make these molecules ready to go. But what if you want to do the imaging of neural activity or other signals in general? Well, unlike optogenetics, You can't just find a molecule in the natural world that's ready to go, right? But the good news is many great protein engineers have made proteins that can help. You can take the green fluorescent protein from jellyfish or red fluorescent proteins from coral and fuse them to some kind of binding domain. Take a calcium binding protein and connect it to the green fluorescent protein just right, and you can make a fluorescent calcium sensor. And so on. You can take all manner of binding proteins and connect them to fluorescent proteins, and you can make sensors of cyclic AMP, protein kinase A, the list goes on and on. This is a screenshot from Jin Zhang's database, Biosensor DB, from her group at UC San Diego. Hundreds of fluorescent sensors have been made. So we started wondering, I think we've all seen cartoons like this. It's a cell, and cells take in many kinds of inputs, shown as the little ovals on the left, growth factors, hormones, and nutrients. Inside the cell is this computational network. Each oval is a different protein, and each arrow is a different interaction. And then finally, the cell will make a decision. Maybe it lives or dies. Maybe it moves. Now, each arrow might have been a different Ph.D. thesis worth of work. And each arrow might have been characterized in a different cell type. So how do we really know what this network looks like? Well, we decided to try to build ways of imaging many things at once in the same living cell. And we developed two ideas, one called spatial multiplexing and one called temporal multiplexing. Basically, these are how the internet works, right? Or how cell phones work. So spatial multiplexing, if you walk around a building, your cell phone will be picked up from one antenna and then another. And so we wondered, what if different places in a living cell could tell you different stories? Then you could measure many signals in the same cell, right? And then temporal multiplexing, that's how radios and other devices work. If they, when you beam a radio signal across a city, or if cell phones are all making calls from the same room, the way it works is that they have different orthogonal signals. And that way they can be sorted out. Let's talk about how it works. Let's do spatial multiplexing first. When Shannon Johnson and Chengyang Liu were working in my group, we worked out how to do it. Suppose the points labeled 1 could have clusters of those reporters I mentioned earlier, maybe of one kind, a calcium reporter. The points labeled 2 would have clusters of a different reporter, like a protein kinase A reporter. While the cell's alive, each dot will blink out a little movie of the signals that are found there. And then you can preserve the cell and stain an image using techniques like I described in the first half of the talk to figure out which sensor was where. Here's how we made it work. Take different fluorescent reporters off the shelf made by others and fuse them to different self-assembling peptides. These are peptides that self-assemble, kind of like the coat proteins of viruses, into clusters. Also, each reporter is linked to a different epitope. Then you can stain it and figure out which reporter is in each puncta. So here's a hippocampal neuron in culture expressing 3 sensors: calcium reporter GCAMP6, cyclic AMP reporter CAMP-R, protein kinase A reporter X-ray ACAR. The green images are what you would see while the cell is alive, and the false color images are what you would see when the cell is preserved. So you can see that there are different clusters, and they can then be identified through staining with antibodies against the epitopes. But while the cell's alive, you can be imaging many kinds of signal at the same time. Here we're giving a drug forskolin that makes cyclic AMP, and then we're imaging cyclic AMP, calcium, and pKa. It's important to see all 3 at once because that's how you can see the relationship between them. In panel H, for example, some neurons had slow calcium, and in panel I, some neurons had fast calcium. In panel L, we can see the relationship. The slow calcium neurons of H had lower pKa, and the faster calcium neurons of I had stronger pKa. Let me say that again, just to make sure I get it right. The slow calcium neurons of H had weaker pKa in L, and the faster calcium neurons of I had stronger pKa in L. So we can relate the speed of one signal and the amplitude of another. This would be hard to do if you image the signals in different cells, right? We can add a fourth indicator, a protein kinase C indicator, and see 4 signals at once. Or we can add a fifth indicator, an ERK kinase indicator, and see 5 signals at once. So that's great if you have sensors ready to go, but what if you don't? What if you just want to know whether a gene is on or off? Or what if you want to know where a protein is in a cell? And so we tried to make basically the molecular equivalent of the AM radio. Okay, this is work done by Yong Chen when he was a postdoc in our group. We used RSFPs, reversibly switchable fluorescent proteins. For example, a green one, if you shine blue light, will glow green, but it will go dim. It's reversible though. Shine purple light, it'll get bright. Here are 6 different greenish proteins with different kinetics. They have different exponential decays under blue light illumination for a few seconds. Again, the frequently asked questions. This is not photobleaching. It's not fluorescence lifetime. It's photo switching, a reversible process. So if you have a bunch of orthogonal signals, the same way that many AM radio stations can work in the same city, or many cell phones can operate in the same room, then you can do linear unmixing and sort them out. I can walk through that a little bit more slowly here. Here we have 3 cells. Each pixel have different RSFPs with different kinetics. Take a brief movie, and every pixel will be a linear combination of the exponentials associated with those RSFPs. Linearly unmix, and you can recover all the original signals. So again, here are the 6 different ones that we use for the green. In panel C, in the upper right, we linked each of these 6 proteins to a different set of skeletal or organelle protein, endoplasmic reticulum, tubulin, lysosome, so forth. Take a brief movie, unmix, and we can get all 6 parts of the cell. That's structure. What about dynamics? We can look at the cell cycle. Here are 4 proteins that change at different phases of the cell cycle. We linked each one to a different protein that glows, but also has this flickering at different rates. Take a brief movie, and we can invert each movie into 4 pictures. So here's a brief movie at time t equals 0, another movie around 5 hours, another movie around 6 hours and 45 minutes. Anyway, we can take many, many movies, invert each into a set of pictures, and image the cell cycle with a 1-color microscope. Here, just for fun, we added in 2 more indicators, seeing 6 things at the same time. And we were able to actually map out some parts of the cell cycle and find signals that people did not know at the time. So to summarize the third technology part of the talk, we found that we can take ideas from telecommunications and make the chemical versions thereof, spatial multiplexing and temporal multiplexing. And by doing so, we can image a potentially unlimited number of signals in the same living cell. We share all of our tools freely, but lately we've also started thinking to use them. What if we could image lots of signals using our imaging tools, perturb them with optogenetics, and then map the structure with expansion? Could you make a computer model of a cell or even a whole brain? Well, we thought small brains might be a good place to start. When Limor Freifeld was in our group, we showed we could expand the larval zebrafish brain. JU showed we could expand the worm skelegans. And lately, we've also been trying to do live imaging of all the brain activity of these species. Here's work by Ziguang Wang and Jack Zhang, working with my group and Matt Wilson's group. We built a very fast microscope to try to image voltage across the whole zebrafish brain. And we could image the voltage of about 25,000 neurons distributed around the zebrafish brain. The brain is in the upper left. You see all these electrical looking traces in the right, but they're not electrical. They're being imaged on a microscope. So how do you put it together? Well, we'd love to model the brain. What if we could collect lots of data while brains are alive in the upper left, and then make molecular maps in the lower left? We can then try to infer what the network does. We could try to understand the underlying physiology, and then bottom up, we could try to predict the physiology for the molecules. So working with Conrad Cording and many others, we're trying to put together this workflow. What about bigger brains? Well, the people use zebrafish and C. elegans because they're small and transparent. Mammalian brains are big and opaque. So one thing we're trying to do is make the living brain more transparent. In work with Seer's group, led by Giovanni Tali-Francesi and Ishan Gupta, we showed that by taking pieces of the mouse brain and adding a safe FDA-approved molecule, dextran, we can make the brain more transparent. Basically, we're evening out the flow of light in the brain. The brain scatters light because lipids and water have different refractive indices. If we raise the refractive index of the water a bit, we can smooth out the flow of light. So in panel D, on the left, you can see a piece of the brain with some beads behind it. On the right-hand side, after we add dextran, the beads are more clear. Here's the live mouse brain. If you just focus where my mouse is pointing, we can zoom deeper and deeper into the mouse brain using 2-photon microscopes, but the neurons become dim. If we add dextran, though, it looks brighter. Importantly, the neural codes were not changed. We want this to be a safe process. So to summarize the last part of the talk, we love to work using these tools ourselves, integrating them into a single workflow, imaging activity, perturbing activity, and finally expanding to make a map. We think those data sets would be enough to give pretty good models of how living things might work. I think I've acknowledged along the way all the people who led different projects, but at the top are a long list of other alumni and current group members who helped. And in the middle, a long list of just the professors leading different groups all over the world who we work with. Please visit our website, synthneuro.org, and you can always email me at my email at boyden@mit.edu, and I'm happy to take some questions.
Thank you so much, Dr. Boyden, for such an engaging lecture. We have some questions that came in through the chat and also as a team that we wanted to ask you. The first is, expansion microscopy has opened up a frontier in structural biology, but as you mentioned, it is inherently destructive. And so do you envision a future where some form of expansion microscopy or microscopy-inspired strategy could be adapted to minimally perturb or even live imaging contexts such as organoids or dynamic tissue slices? Or is its true potential fundamentally tied to postmortem fixed material?
I don't see how it would ever really work on a fully living thing, but if you can make a map of the molecules and how they interact, that's a pretty good data set that would tell you how, molecules work together, right? So I think if you have, one of the big themes of biology, right, is that if you have good enough structure, you can make some pretty good inferences about function, right? AlphaFold for protein structure, the sequence of the human genome, those are all static structural things, right? But look how much you could get out of them, right? What we lack are the structures of cells and tissues, and that's what I hope expansion can give.
That makes a lot of sense. Thank you. Do you want to ask the next question?
Yeah. Should we go to the chat or I'll go here. This one. Okay, so our next question that we've generated as a team is so this is a little bit less technical of a question, but yeah, we were thinking about this as you were talking. Your work really elegantly bridges the molecule to the system, but neuroscience still really struggles with the question that is, will full understanding come from bottom-up reconstruction from molecules to networks? Or do you think we need to discover emergent principles at higher levels of abstraction? How do you think about this tension in your own tool development?
I'm not sure levels of abstraction exist. That's a computer science concept, and in computer science they exist because humans designed computers that way, right? In the old days, you might need to actually know what zeros and ones are doing in the computer to program it. Now, you can use a high-level programming language, or frankly, nowadays people just talk to ChatGPT. But that's because humans designed it that way, right? What level of abstraction means you could ignore certain details. How do you know what you can ignore in the brain? We don't know for sure, right? And so I think that's why the concept of levels of abstraction can sometimes be dangerous, right? Almost every time we've ignored something in biology, there's been some reason to regret it later, right? Some weird mechanism kicks in and and you want to know why it happened, right? So that's why I want to solve the system completely from atoms to entire thing, right? All the parts and how they work together. And if other people can solve biology without doing that, that's great, right? I will go lie on the beach and watch the ocean or something. But it doesn't seem to be happening yet. So we're going to keep making tools and giving people the tools, and so people can go from the smallest scales to the largest scales, like across temporal scales, and deal with heterogeneity.
Thank you so much for that answer. The next question that's come in is, your lab has produced so many paradigm shifting tools from optogenetics to expansion microscopy. How do you personally stay creatively generative amidst all the pressure for deliverables? And how do you foster a culture of curiosity and innovation in such a high output environment?
Well, I guess those are two separate questions. I really do believe that creativity is learnable and teachable. I wrote two essays out of Marblestone, one called Assumption Proof Brain Mapping and one called Architected Discovery, which describe our methods. So I do think creativity can be learned and taught. I mean, the other question is how to do well in the business side of academia. I've always considered myself to be pretty bad at fundraising. The vast majority of our grants get rejected. That's tough. I don't have a good answer for that.
And then another question that's come in is, so looking back at some of the most transformative technologies you've developed, what do you think made them succeed? Do you think it was their novelty, their simplicity, their timing, or something else? And now when you're evaluating new ideas, what criteria do you look for to tell whether they're worth investing in, maybe based on your previous experience?
Well, I think the most important thing is, are we getting to the ground truth, the fundamental building blocks and how they work together? And if the technology works and it's easy to use, then that's the criteria, right? And you can figure that out just by looking at the history of biology, right? The history of biology shows that this is what matters.
Thank you so much. Another question. Optogenetics, sorry.
Wait, sorry.
Okay. Optogenetics has given us an unprecedented ability to casually perturb the brain, but manipulating activity doesn't necessarily translate into understanding thought or consciousness. How do you personally conceptualize the gap between circuit level causality and high level cognitive understanding? Do you think new tools will close that gap, or do we need new frameworks entirely?
Well, the problem with consciousness is we can't measure it. I really do want to figure out how to measure consciousness, because if you can't measure it, how can you study it? Um, yeah, I think that being able to measure is really key. So yeah, we need to develop that strategy if we want to connect it to the rest.
That makes sense.
And then one question that just came through from one of our participants was, could we use expansion microscopy to create patient-specific digital twins for predictive modeling and clinical research? I guess more of a technical question.
How would I mean, expansions make something bigger? How would that make a digital twin? I didn't quite understand the question.
Yeah, the question, and maybe whoever wrote this can.
Yeah, I heard all the words of the question. I just don't know what the question
Yeah,
is.
maybe El Mubarak, if you want to elaborate on your question. But I think for now too, we asked our previous keynote speaker this: if you have any like advice for an undergraduate, maybe looking to go into this field, like what skills we should work on developing. I know, yeah, most of us on this call are undergraduates, and a lot of us are working in labs. So, what your advice is? What would your advice be to undergraduates who, yeah, are going through college right now and are maybe working in labs, especially in like this new era with like AI? What skills you think we should develop and what we should focus on as we move towards our careers?
I'm very bad at general advice because the opposite of good advice could be better advice. I guess the better advice I would give is whatever advice you hear, visualize what would happen if you did the opposite and see if you like that better. But I do think knowing the fundamentals is really good. I feel lucky that I was an undergrad for 6 years, so I got to learn physics and chemistry and computer science and electrical engineering, and I think I'm one one class short of a math major degree as well. So I have to learn, almost all the fundamental sciences, right? And that's great because 25 years later, those are the sciences where they're still true and we still know what we're doing. So yeah, I think that knowing the fundamental sciences really in a diehard way is very helpful. So that's what I would recommend to undergrads. And then um, yeah, I'm working on an essay right now on like what are the key things to know because not everybody has the luxury to be an undergrad for 6 years, and so I did I'm working with a couple students now to make sort of the short curriculum, and we'll post it on the web probably this week or next week. What's the minimum stuff that you need to know to be really ground truth oriented, revolutionary in innovation.
That sounds amazing. Thank you so much. It is 1 o'clock, so we're gonna break now. Thank you so much, Dr. Boyden, for your time and for your engaging lecture. I know our team and panel and attendees really enjoyed hearing your talk.
Have a
Thank you
great
so
day.
much.
And to all the attendees, we will return at 1:15 for our panel with the Harvard Grid Accelerator.
All righty. I think we're all set here. Great. Welcome, everyone, to our afternoon panel session at Frontiers of Biotechnology. And we're thrilled to be joined by a remarkable group of scientists and innovators from the Harvard Grid Accelerator, an initiative that's accelerating the translation of early-stage life science discoveries into real-world therapeutics. This panel will spotlight the cutting-edge projects emerging from the program and the individuals leading the charge in fields like neurorehabilitation, sensory augmentation, and machine learning-enabled diagnostics. So panelists, welcome. As we've discussed a little bit, we'll have this in a lightning pitch format. Each of our panelists will give their talk, we'll ask a couple of questions, and then go on to the next. So before I introduce each of you guys, we're honored to be moderated by Ethan Schaefer, Senior Analyst at Ingrail, and I'll let him introduce himself briefly beforehand.
Yep. Hi, nice to meet you guys. Excited to hear all your pitches. I am only a few years out of college myself, and I kind of work at the intersection of science and commercialization. I'm pitching new company ideas to venture groups all the time, so hopefully I'm able to give you guys some good feedback on these pitches.
And yeah, I think Ethan's quite humble. He brings a strategic lens to biotech innovation, working at the intersection. And yeah, at Ingrail, he does a wonderful job identifying and communicating commercial potential of novel treatments. And so thought he would make a perfect guide for today's conversation. And yeah, so let's get into it. I'll introduce you guys. So first, I will have Santiago Riaz, who's worked with Professor Connor Walsh, focuses on stroke rehabilitation and the restoration of motor function through wearable robotics. Then we'll have Haritosh, a postdoc in Joanna Eisenberg's lab, who is engineering portable breath-based diagnostics for disease detection, combining smart chemistry with machine learning to build accessible, scalable solutions. And finally, we'll have Raymond Liu, a CS PhD candidate in Patrick Slade's lab, whose work integrates hardware and AI to build next-gen exoskeleton systems for restoring mobility. Cool. With that, let's start with Santiago.
Hello, everyone. Can you hear me? Sounds good. Okay. Can you see my screen?
Yep.
Okay. All right. So yeah, I'm Santiago Canete. I work in Connor Walsh's lab. I also actually work in Patrick Slade, so I'm co-advised. But anyway, this is Stride Bionics, and our mission is to redefine human potential through wearable technologies. Okay. So the first application that we're targeting is the mobility impairment that comes after a stroke. Stroke is the leading cause of disability worldwide. It affects 60 million people worldwide, 8 million in the US. There's 800,000 new cases every year, and 70% of these people suffer long-term impairment. which ultimately affects their quality of life. It causes inactivity, which leads to other health effects, increases their healthcare cost. So overall, this results in a direct and indirect cost to the US of around $150 billion a year. So more specifically, one of the things that happens after a stroke, people lose mobility in half of their body, and a large portion of this effect is at the ankle. So this leads to ankle weakness, where people are at a higher risk of falling. They cannot propel their body forward as they move. And the current solutions are really limited. They basically use plastic boots that are called ankle foot orthosis, or AFOs, which basically lock the ankle in place and avoid the toes dropping as the person walks. This solution is really not liked very much by clinicians or patients, with only around 30% of people using them long term. And it really encourages independence through compensation, since people are not able to move their ankle as they walk. Clinicians like Dr. Breen, who have been working in this space with stroke patients for around 30 years, who's also the lead, the director of rehabilitation at Whittier Hospital, thinks that their solutions are really limited. And basically, they only prescribe AFOs because it's the only thing they have to allow people to return home safely. But in the last few years, there's been a new landscape opening up for assistive and rehabilitation robots, with several robots for different conditions getting CMS coverage for different amounts. So in the last few years at Harvard, our team has been developing a similar concept to the AFOs, but this version of it is intelligent. It can adapt to the user's needs and provide active assistance through motors and sensors. So it can help the person lift their toes up as they walk and also propel their body forward. It can increase the person's force at the ankle in around 40%. It's pretty lightweight, below 2 pounds, and it can allow someone to walk for over 2 hours. This also integrates a set of wearable sensors that the clinician and patient can use to track the recovery and inform therapy. It also uses machine learning algorithms to personalize assistance to the user to target a specific clinical outcome. So currently in the research space, we have tested this device with around 20 patients that have really liked the device and seen significant benefit to their walking. And we've also shown that there's a significant clinical effect, almost immediate, when a person wears this device, where they're able to increase their walking speed and their gait quality, which is basically an aggregate measure of gait symmetry and compensation strategies. So we plan to commercialize this device and launch a startup from our lab, which is going to be, our go-to-market strategy is going to follow 3 stages. In the first stage, we're going to be focused on developing the device with clinics and patients in the loop, so that we can ensure that we're building a device that people want to use and it's effective. This will lead us to the first stage, which is basically FDA clearance. And from there, we'll focus on acquiring more clinics through key opinion leaders, conferences, etc. And ultimately, the overall goal is to achieve Medicare reimbursement for this device. So we can launch a device that can be used independently at home and it can get covered, basically. All right, so our solution is composed by 2 aspects. The one is the hardware, where following how basically this space works today, where a person would get a device prescribed by a clinician and then fitted by an orthotist. We plan to use these existing workflows to get our hardware to people's hands. And on the other hand, we have the software solution, where people would be able to track the recovery, and the therapist can see how the person is doing and inform therapy remotely, since basically once a person is discharged from outpatient care, they only get to see their doctor maybe once a year or so. Okay, so this gives us access to a $6 billion market in the orthotics, where basically this is calculated given the number of stroke or patients that could benefit from this current device times the price point that we have set for our device. And in later stages, we plan to expand to the overall market, like covering other conditions and more of the home health services space. We believe that this is the right moment to do this, and given the changes in the regulatory and reimbursement landscape that now, or CMS now sees the benefit for this device, and there's a clear path towards getting these devices reimbursed. We also think that it is the right time in the clinical space, since the cost of rehab for stroke just keeps increasing. There's been a shift towards telerehabilitation and at-home rehabilitation. And clinics continue to try to incorporate more technology into their day-to-day lives. We also believe we have the right team to do this. I myself hold a PhD in robotic rehabilitation with a focus in control and biomechanics. I've been working on this for over 10 years. And then we have Miles Cooper, who is a very good mechanical designer, a PhD student also in the lab. He's very good at developing very wearable and portable actuation systems. And recently, Gabe Valdez has joined us, who has a lot of experience in the business side of things, and he's helping us develop this go-to-market strategy and form basically all the business case for our device. We're also, in our team, we have Dr. Colonel Walsh and Patrick Slade, who have a very long track record of publications and very impactful research in this space with both clinical populations and healthy populations. And we're backed also by a large team of commercialization experts, clinical partners, medical device experts, and regulatory and reimbursement consultants. Okay, so right now we're looking in the short term to raise around 600,000. This will allow us to expand our team with 2 engineers, perform all the customer validation we want to do with the clinics and patients in the loop. So fund all the R&D and product development, etc. And this would be an effort for the next 6 or 8 months, where we plan to sign 2 partnerships with 2 clinics, have one signed a letter of interest to place a purchasing order by the end of this partnership. And we plan to send 4 devices with patients for them to use them independently at home for about 2 months. And this will lead us to the first FDA pre-submission, basically. In the next stages, we're looking to raise $7 million after that, once we have gathered all the customer validation and we've proven that clinics and patients are interested in this technology. And if I can leave you with one thing, is that mobility impairment should not limit our independence. Thank you.
Thanks so much, Santiago. That's a really compelling case. I think what stood out to me most there was, the data that you guys have generated already looks, striking. And really, actually, another thing that stood out to me was the difference in cost relative to some of the other exoskeleton approaches. I think that could be really advantageous as you move towards commercialization. I did have a question around Medicare reimbursement. My understanding is that the post-stroke rehabilitation market is primarily Medicare, I would think. So CMS reimbursement is key there. What is the level of evidence that you guys are looking to generate in order to obtain Medicare reimbursement?
Yeah, so obtaining Medicare reimbursement is kind of a long-term thing where you have to show for a few years that people really want your device, that you have that clinical evidence where people's lives are basically better, and also that you are actually saving money to the insurance. So because there's that gap between when you get FDA clearance to basically commercialize your product and when you get medical reimbursement, what we plan to do is through basically acquiring clinics so that people can start using in clinic. So basically that will be our initial market, and then there we start gathering that evidence of clinical benefit and patient interest, and then just building a case that if we can show that people are more active using their device, that their recovery outcomes are better, that ultimately will save a lot of money in all these healthcare related costs after a stroke. Yeah.
Awesome.
Okay, thanks. Sorry,
Cool.
Sean, go ahead, please.
Yeah, thank you, Santi. I had another question as well, and this is back to one of the first slides that you presented, You noted that while the AFOs are often prescribed after stroke, only about 30% of patients do continue using them long term. And that's a pretty striking drop off. So I would love to better understand what tends to happen with the other 70%. You touched on this a little bit, but is it discomfort, lack of effectiveness, something else driving that discontinuation? And as you think about the reimbursement, would patients typically have to demonstrate non-response to this traditional AFO before qualifying for coverage of your exo device? How is that, pipeline working?
Yeah, so I mean, in terms of the what happens with AFOs currently, it's mostly people don't like them because they're uncomfortable, right? Like it is hot to wear it is heavy, it feels restrictive, right? Because ultimately it's just locking your ankle in one position. And clinicians don't like it because basically when you have your ankle locked, you are not able to use it. So ultimately it requires people to adapt their walking to use other joints. So it's a very inefficient way of walking. And the belief is that it just hinders recovery overall. And then, so basically the precedence in terms of what has happened with other devices, like for example, powered knee prosthesis. Like what happens is when a better technology comes in, it basically replaces the previous one. So if like in prosthesis, it used to be that a knee prosthetic was like around $6,000. When the power needs came in, all of a sudden knee prosthesis were like $26,000. And for Medicare, that is a good approach, paying for that, because basically it just makes people's life better and reduces their cost, but it also like multiplies a market by 4. So like we see this as a way of replacing ankle foot orthosis, right? Like that would be the ideal scenario. Obviously, it's a very patient-to-patient thing. So it would ultimately also be decided by the clinician and the orthotist what the best approach is. But if there's a case where a patient meets all of the Medicare criteria, then there's no reason to pay for an AFO.
Understood. Thank you.
So I know we're short on time here, but I did want to ask you one more thing, kind of along the line of the path to obtaining Medicare reimbursement. Have you guys considered, is there a path where you could potentially get commercial reimbursement more quickly than Medicare? Because, the CMS moves very slowly.
Yeah.
And with that pilot data you're showing, I mean, that's certainly compelling, particularly for patients that can't tolerate or AFOs just aren't working for them. If they're going to otherwise progress to a $70,000 exoskeleton, there seems to be a place for this.
Sorry, what was the original question?
Have you guys considered a path
Oh, yeah, okay.
for commercial
Yeah.
reimbursement initially?
Yeah, so I mean, in that gap basically between FDA and Medicare reimbursement, the plan is to go into like integrated healthcare systems, basically like the VA. There's another few that we plan to talk to. We started to talk to the VA, and basically that is like the easiest and fastest path because they're always very interested in new technologies and paying them for their patients. And then I think the clinics here play a very important role in between FDA and reimbursement, where we would sell devices to clinics. Then there's obviously some early adopters or evangelists that would be willing to pay for the device. But yes, it's a long-term process where until we really would break even, and that will happen after we get Medicare.
Okay, thanks.
Yeah, I don't know if that answers the
No, I understand, dealing with payers, it can be, it's a real like push and pull balance. But yeah, this looks like a really promising idea. So I truly wish you guys best of luck.
Yeah, thank you.
Thank you, Santiago. Cool. Let's move on to Haritosh. Would you like to present on SpiroSniff?
Perfect. I'll start sharing my screen.
Okay. So it works.
Sweet. Sorry. Are you able to see the screen?
Yep.
Yep.
Perfect. Thank you. Well, thanks for inviting me to speak on SpireSniff, a ML-driven breathalyzer for lung cancer detection. And I'm speaking on behalf of a full team from Joanne Eisenberg's lab that is developing a VOC detector that can allow us for various different disease detections. The one we're focusing on right now is breath analysis for lung cancer. So just to give some background, there's about 20% of cancers that are in the United States related to lung cancer. So that's a huge pool of population, which really amounts to a huge expenditure of about $24 billion just for treatment and diagnostics alone of lung cancer. One of the more staggering statistics around this is not really the cost of treatment or diagnosis of lung cancer, but really the disparity between who's eligible to be screened and who actually is screened. And about only 6% of those who should be screened or are eligible to be screened are actually getting screened for lung cancer. And this really causes a huge downward cascade between treatment and between early treatment. And one of the standard methods and why screening is so difficult is because it requires a lot of invasive and costly procedures where the patient has to go to the hospital and they have to both get a radiology screening, but also a biopsy, which takes a lot of time and cost out of the patient's lives. Usually, most of the times you might think you're healthy, so you might not regularly be doing this. So this is really where the disparity is coming from. There are some new tests that are emerging, like the Galleri test, that is a blood test for at-home cases, where people can send out their blood sample. Of course, you still need a technician to draw that blood sample, but it's a bit more easier and accessible. And it does detect over 50 types of cancers, according to their data sheets and their claims. And they do have 95% specificity, which would be ideal. But there are actually huge downsides to a test like this, where actually that specificity and sensitivity is only applicable for late stage lung cancers. But really, if you look at early stage or stage 1 lung cancer sensitivity, it's only about 17%, which is really where we want to tackle detection of early diagnostics and treatment. And also, a singular test like this can cost upward of about $1,000. So if you were to do it almost monthly or every year, it could really increase the cost. So really, we saw a problem here where there is no current lung cancer diagnostics that are accessible and non-invasive. So we really turned to a new method that has started to emerge called detection from breath analysis. And it's really because we've started to characterize in the last decade that there are specific volatile organic compound markers that are coming and emanating from the exhaled breath of unhealthy patients that is slightly different than the healthy patients. And the characterization of these particular signatures for specific diseases is really what's causing a huge boom in this type of technology. Really, for example, in lung cancer, these 5 compounds, these 5 VOCs, have been seen in different proportions for lung cancer patients, always elevated compared to the healthy patient controls, which serves as a great chemical biomarker for us to detect from exhaled air. So really, SpireSniff solution is quite simple. Just like any alcohol breathalyzer you might see on a car stop, you have an at-home device where you can breathe into this breathalyzer, and we have a series of VOC sensors on an integrated printed circuit board that detects these VOCs and feeds it into a machine learning model, which then allows us to predict a likelihood assessment of lung cancer based on these proportions and mixtures of those particular compounds. And so just to give you some insights of some of the development we've done, we've of course made the housing and prototype of our breath analyzer fully integrated with VOC sensors and battery inside, and also a audible and visual display for users to understand how to use it. When we were doing lab testing, so what we essentially did was we took 4 of these chemicals, VOC chemicals that were related to lung cancer patients, and we made them into proportions of healthy and unhealthy concentrations, and we were able to get remarkable distinction from our VOC sensors in an ability to discern these specific time response curves. The importance of time response curves is actually quite important in chemical sensing. It's because if you were just simply looking for static signals, you might only get a baseline value of how much VOCs are there. But usually the chemical identity and the way they transport through the device and the way they absorb and desorb onto the device really gives a huge amount of information of what chemicals are there and how much. This is really driven by bioinspiration of how dogs sniff the chemicals, and really we're mimicking that for our device here. When we feed this into a machine learning model, we actually get quite remarkable accuracy, about 92% for the ROC curve, and the sensitivity that was about 17% for the Galleri test, we actually can get up to approximately 88%. So this has a huge improvement from already standard of care at-home kits. And of course, there's a lot more iteration, improvement, and testing to be done, but this is a great step forward for SpiroSniff. So we are looking to commercialize this device and this technology at large. And at first, we are really looking to the lung cancer market, where either we would be looking to sell to hospitals or clinicians themselves, in which they want to do quick pre-screening methods, or also prescribing these devices to patients at risk. Those patients at risk could be ones that have either been cured of lung cancer and they might undergo remission, so they might be able to track that and monitor that from home, or also those with already predispositioned histories of lung cancer where they should be tracking it after a certain age. After this, we look to expand into other cancer markets, so including breast cancer, which has also been cataloged for specific VOCs, and also malaria and so forth. We have a pretty diverse team and a set of core team members that just graduated from their undergraduate programs that are looking to spin this out, along with scientific advisors like myself and Joanna Eisenberg and Dr. Jacopo Mobili, who's a biosensor expert, along with business advisors from Harvard Innovation Lab that is helping drive our technology forward. Thank you for your time, and I look forward to hearing your questions.
Awesome. Thank you so much. Well, I'll jump into questions. I mean, this is, a fascinating project. And you jumped the gun a little bit on the question that I was going to ask about VOCs and the other possible indications that this could be applied to, starting with lung cancer, but you mentioned malaria as well, other types of cancers, breast cancer. Yeah, I guess, is there evidence for other diseases beyond the ones that you mentioned? How sort of like widespread can we be thinking about the use of VOCs generally? And also, well, I guess let's tackle that one first, and then I can ask a follow-up question about the diagnostics platform in general.
Absolutely. So right now, 3 major categories of illnesses and diseases are being tracked for and cataloged. So in the cancer space, lung cancer and breast cancer and pancreatic cancer are 3 types of cancers that are trying to be cataloged for the VOC signatures that they're emitting. Really, it's simply because of the metabolites you can sense from the exhaled air. 2 other areas that are being investigated is, of course, malaria, but one more is neurodegenerative diseases like Alzheimer's and Parkinson's, which also have seen different signatures of VOCs. But one thing we do have to be careful is, one of the things we're noticing, of course, is this early research that's happening in the space. So one of the markers, for example, for lung cancer was toluene. It's a compound that's quite prominently found in lung cancer patients. But also, it could be a false marker that we might be tracking, because actually a lot of smokers have a lot of toluene in their breath. So it might just be that we're tracking who's smoking and not smoking rather than lung cancer. So really, systematic studies that are still being done now, we can't say they're all complete, are being mapped out for these different VOCs. But this device really is flexible in a sense that once you have the right sensors and the right ML model trained, you can really capture any of these diseases once it's completely mapped out.
Cool. And yeah, just a quick follow-up, right? As the field is moving towards understanding VOCs, and building out similar sort of like breath analyzers. How are you thinking about your unique approach and what stands out about SpiroSniff specifically?
Yeah, so I did brush this over quickly, but one of the unique things about SpiroSniff and the underlying technology, actually, which we call eye noses at Joanna's lab, is intelligent nature-inspired olfactory sensors engineered to sniff. It really derives from bioinspiration. We were really turned to the dogs and how they are capable of smelling compounds down to parts per billion or parts per trillion, even from miles away. And they're, indispensable for applications of search and rescue. Even, you've probably heard dogs also sniff out malaria. So they're all over the news. And really, when we looked at their biology, we learned a couple of important things. The first thing which everyone knows is, okay, they have better olfactory receptors that might help detect it. But that's not everything. Of course, the hardware matters, but it's the strategies they use to uptake the air and modulate it. So you've probably noticed that dogs, they do something called sniffing. They sniff a lot, and they actually modulate how they sniff based on what's being sensed. So imagine once in a single sniff, they get some information, but when they're not confident about the information they're getting, they modulate to another sniff. So allowing the hardware to see different perspectives of the same analyte. And in this same way, this device really isn't meant of a breathalyzer of a single breath. Once the single breath is input into the system, after about 30 to 40 seconds, it actually tells the user to breathe in a different way into the system to get higher specificity and sensitivity without actually requiring very selective sensors. And so we really are, this is the edge that SpiroSniff has over other commercial sensors.
Awesome. Thank you for elaborating, and I apologize for missing the nuance during the pitch.
Oh, no worries.
Ethan, did you have a question?
Yeah, Heritage, thank you so much for sharing. This is a really cool idea, and I think very timely as well. We're starting to see, I think, within the oncology community, a shift towards, we need a screening tool before we're going to go ahead and progress to radiology. Radiology is incredibly expensive, and for high risk individuals, I think a lot of people have trouble affording uh, uh, diagnostics like this. So this is definitely addressing an important unmet need. My question was more on the business side. You had mentioned that this would be marketed as an at-home screening tool, but I actually see I could envision an opportunity for this even in like a primary care setting. I imagine it doesn't cost money to breathe into this tool. Is that
Mhm.
something that you guys have considered?
Actually, this is something we're continuously reevaluating. As you mentioned, it's something our penetration strategy is really being almost forged as we talk in the next year. So once we validate this technology, we're really seeing where the best application was. And we did see at-home use as one of our initial targets, but actually our business advisors at HI Labs exactly said what you mentioned is it might be a technology that is at the primary care physician level, and you are just pitching to them where this technology is adopted by most physicians as an early start to their differential diagnostics method. So I do see it going that way as well.
Yeah, you guys have some exciting stuff to evaluate. I certainly could think that, I mean, penetrating the primary care market generally is a really tough thing to do. But this is a technology that has almost no time burden on the clinician. So there's a real
Hmm.
opportunity here for something like that. So best of luck to
Thank
you
you.
guys.
Thank you so much.
Okay, fantastic. Thank you again, Haritosh. And last but not least, Raymond.
Hey, can you hear me?
Yes.
Yep.
Great. Okay. Okay. And can you see my slides?
Yes.
Yes.
Okay, great. So my name is Raymond. I am a first year computer science PhD student in the Harvard Slade Lab, where I've been working on developing a smartphone-based navigation assistant to help people who are blind or visually impaired. And so globally, roughly 340 million people have blindness or visual impairment, which I'll refer to as BVI, which makes basic day-to-day navigation a potentially dangerous experience. And in fact, 94% of people with BVI have experienced head-level accidents that often result in medical consequences, such as hospitalization or visits to the emergency room. And this reduces people's confidence in their ability to navigate independently. And in fact, 40% of people with BVI walked new routes less frequently than once per week. BVI is also associated with a variety of negative outcomes, such as lower educational status, lower income, unemployment, reduced physical and mental health, and reduced quality of life. And it is a leading cause of a need for an assistance with performing routine activities, such as grocery shopping or working at a job. And so assistive technology to help people with BVI navigate independently could improve physical and mental health, quality of life, and socioeconomic status. And so we conducted a survey of 99 people with BVI to determine what capabilities an ideal navigation aid should have, and we identified 3 major capabilities. First is obstacle avoidance, which helps users avoid potentially dangerous collisions. Second is outdoor route planning, which allows users to follow turn-by-turn directions to specific destinations. And third is path guidance and orientation to help people stay oriented while navigating along a desired route. And so there are many existing approaches. The most common ones are standard white canes, guide dogs, electronic mobility aids such as smart canes, and various smartphone navigation apps. However, these tend to be limited in either capabilities or accessibility. So a white cane is simple, it's low cost, and it's useful for obtaining haptic information about the surface of the ground and nearby obstacles, but it can't detect obstacles beyond the length of the cane and is less useful for higher obstacles such as hanging tree branches. Guide dogs can detect obstacles and provide path guidance, but can only navigate along known routes. And additionally, only about 2% of people with BVI own a guide dog because they are expensive and time-consuming for the owner to take care of, as well as for organizations to raise and train. There are some electronic travel aids on the market, but each one typically has only 1 or 2 of the key navigation capabilities that we want, and those are most often obstacle detection or turn-by-turn navigation. And these also tend to be very expensive, ranging from $800 on the low end to upwards of maybe around $5,000. Now, smartphone-based solutions are promising because smartphones are ubiquitous, they offer easy deployment of software, and they contain a variety of software, a variety of sensors, and they are small and lightweight. And also, many people with BVI use smartphone-based solutions because they are so accessible. But existing smartphone navigation apps such as Google Maps, as well as navigation apps created specifically for people with BVI, such as Blind Square and Soundscape, only really leverage the smartphone's GPS or compass to perform things like GPS navigation or provide information on nearby landmarks. So bearing in mind the problems with these existing approaches, we've developed a smartphone application that has all 3 of the major navigation capabilities that we laid out. So to perform obstacle detection and avoidance, we use the camera, the IMU, and if the phone has one, the LiDAR. We use GPS and IMU to provide turn-by-turn directions during outdoor navigation. And to provide more detailed orientation information, we trained a lightweight semantic segmentation model to detect key elements of street scenes such as sidewalks, crosswalks, roads, and curbs. And importantly, since our solution is a smartphone app that relies solely on the sensors and processing power in the phone, our system can easily be made widely accessible to many people. And the missing component here is audio feedback, because while the visuals are nice, if you have BVI, you may not be able to see that. And so in order to guide users with BVI, our system provides audio feedback to have them turn towards a specific direction, with this audio feedback consisting of repeated beeping sounds that play spatially to the right or left through open ear headphones, so users can tell where they need to be going. And our system also personalizes this audio feedback to each individual user as they use our system to improve the benefits that they receive. We performed experiments with 14 participants with BVI to evaluate our system and compare it with a standard white cane and Google Maps for outdoor navigation. And we found that with a minimal amount of training, users were able to navigate an outdoor path in 13% less time and experienced a 41% decrease in collisions. And in post-experiment surveys, users reported that our system had a high usability score, a very low workload score relative to Google Maps. Since we intend for our application to be able to provide similar or better navigation capabilities compared to a guide dog, our target serviceable obtainable market is the set of US-based guide dog users, which comprises an estimated 300,000 people. Based on the survey we conducted, people indicated that they would be willing to pay on average up to around $425 for a device with all the navigation capabilities that we identified. And so our application would represent a 98% annual saving for the user compared to owning a guide dog. In terms of our current and future goals, we'd next like to polish our system with additional input from orientation and mobility specialists at community organizations who have a good understanding of the daily navigation needs of people with BVI. And then we'll broadly deploy our system to users for at-home evaluation over a longer period of time, such as 1 month. And to support discovery, we'll work with these community organizations such as the Perkins School for the Blind or the Carroll Center for the Blind, which we've partnered with in the past because blind and visually impaired communities often depend on these organizations for guidance on the best technology. We'll also need a business lead to develop a go-to-market strategy, and we will pursue funding for next steps, for example, through the NSF SBIR program. All right, thank you.
Thanks, Raymond. Yeah, I think, again, definitely an under-recognized unmet need here. One that, frankly, I haven't even fully appreciated. So I appreciate you providing some of that background. I actually had a question on the clinical data that you generated so far. Would you mind skipping back over to that slide?
Yep.
Yeah, so can you just help me understand here? This data looks really good. So I just wanted to better understand what you guys did here. So for these patients, it looks like they're crossing over to the other group. In this study, are they walking the same route twice, or are these in two different routes?
Yes, so we used one specific route with point A and point B. And essentially what we do is patients start at point A initially and then walk back and forth repeatedly with different methods. And we then reverse the order of the methods to eliminate the possible effects of adaptation.
Okay, thanks. And secondly, are you using this technology on top of the white cane, or is this an alternative to a cane?
Yeah, so since white canes do provide physical haptic feedback, which is very useful, and our technology can't provide that, we do intend for people to use a white cane in addition to our system because we can greatly reduce the amount of collisions that people experience, but in the end, there is always this component of physical safety that we are unable to provide simply with just a smartphone application.
Yeah, certainly. So this data that you generated, then this is with your technology alone. This isn't with the white cane in addition.
Yeah, so this is comparing people just walking with a white cane to people walking with a white cane and our system.
Okay.
There's no point where we have people walk without a white cane for safety reasons.
Okay, yeah, good. The 41% improvement, though, I think it speaks for itself.
Yeah.
Sean, did you have a question?
Yeah, I guess I'll just close. Not really a question, just genuine curiosity. What do you think was like the hardest technical or like design challenge that you faced building this? And as you look towards scaling, right, how are you thinking about building that strong foundation, whether through either data, user engagement, IP? Yeah, just how are you and your team thinking about this?
So the hardest technical difficulties we had were with developing something that is both effective but can also work on a variety of different smartphones, because our approach, we definitely heavily focus on accessibility and like eliminating the need for, like, for example, manufacturing requirements. So in using a smartphone-based approach, essentially we have to make sure that our methods both are able to run well on a variety of smartphones. For example, like an iPhone 12 is what we use, which is, I think, maybe like 5 years old now. But it also has to be accurate and effective at the same time. So that was maybe the biggest general challenge. Another one was, yeah, since smartphones maybe have like less effective sensors than if you were to put something together yourself. So like smartphone GPS, for example, typically has errors of up to maybe 5 to 10 meters. In some very bad scenarios, maybe 15 meters. And that makes accurate navigation very difficult for people with BVI. And then to answer the second part of your question, we do in the future plan to file IP on the algorithms we developed to essentially make the system work well on a smartphone. So, yeah.
Awesome. Thank you so much. Cool. Well, I think that wraps up our panel segment. To our three panelists, thank you so much again for your time, for being here. We really, really appreciate it. And I think our attendees have also really appreciated learning about your projects. Ethan, thank you for joining us. It's really good to see you again. And I'll pass it off to Adi and Julia, who will transition us into the poster session. Yeah, thank you all again.
Yep. Thank you to the panelists.
Thank you. Thank you very much.
Thank you.
Thank you again to our panelists. We're now going to move into our poster session where we have some attendees presenting some really, really cool projects. We're going to be doing the poster session on a separate Zoom link in the chat right now. And so if everyone could leave and join that registration link, that would be perfect. So we will see everyone at the other link in the chat right now. Thank you, guys.
Thank you so much to Dr. Liu, Dr. Liaza, Dr. Patel, Sean, Ethan Schaefer, and the Grid Accelerator for such an engaging panel and for sharing the incredible work and incredibly exciting work you do. We also just wanted to take the time to say thank you so much to all of our presenters in this poster session. Adi and I hopped around to a bunch of the rooms and we were just so impressed by the quality of the research and just, yeah, how well rehearsed the presentations were. We were just so impressed. Yeah, so that was great. And yeah, before we wrap up, we just wanted to share our heartfelt gratitude to everyone for being here and for making the Frontiers of Biotechnology Conference such an incredible success. This was our first time doing this over Zoom, and you guys made it awesome. So thank you so much.
We're so grateful for your active participation, whether you asked a thoughtful question during Keynote, joined in during a workshop, or simply showed up with curiosity and openness. You made this event meaningful and memorable, and for that, we're so grateful.
The energy and insight you all brought to the table today truly captured the spirit of this conference. Our speakers and workshop leaders were amazing, and the questions you asked sparked some of the most engaging moments of the day.
And again, like Julia said earlier, to those of you who presented your research, thank you so much for sharing your hard work and your passion with us. It's truly incredible what you guys are doing. We can't wait to see how your research progresses. And to everyone else who contributed in big or small ways, we're so glad that you're part of this growing OpenBio community. And a huge thank you once again to our speakers, workshop leaders, and of course, the incredible team at Harvard Undergraduate OpenBio Laboratory. We hope you leave today feeling inspired, empowered, and ready to keep exploring the frontiers of biotechnology. We hope to see you back here again next year. Please stay connected. Email us at openbiolaboratory@gmail.com for any further questions. And thank you for being here. Have a great rest of your Saturday.
Thank you all.